الأعضاء، إضافة إلى إسهامها في تقديم المشورة العلمية وإجراء الدراسات والأبحاث المتخصصة التي تثري المكتبة التربوية. افتتح نائب رئيس الجامعة للشؤون التعليمية والأكاديمية الأستاذ الدكتور محمد بن صالح النمي الملتقى الأول للوقاية من المخدرات تحت عنوان الوقاية الفكرية. ويأتي هذا الملتقى ضمن مبادرة الملتقيات العلمية نبيه التي أطلقتها وزارة التعليم، وتستهدف المبادرة طلاب وطالبات الجامعة لرفع الوعي لديهم بأضرار المخدرات والمؤثرات العقلية وأساليب إغرائهم بالمخدرات، وذلك للمساهمة في التحصين والحماية من الوقوع في تعاطي المخدرات بين الطلاب والطالبات ومنسوبي الجامعة. إلى هنا وصلنا وإياكم إلى نهاية النشرة، حتى نلقاكم الأسبوع المقبل. تقبلوا تحياتي في أمان الله.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Best wishes to all of us. Welcome to the International Seminar on Plant Protections, organized by the Plant Pests and Disease Department, Faculty of Agriculture, Universitas Brawijaya, Indonesia. In this international seminar, we proudly present remarkable keynote speaker from King Saud University, Saudi Arabia, and Universitas Brawijaya, Indonesia. We bring the themes recent research and advancement in integrated management of plant pests and disease. Now, first of all, in this wonderful occasion, I would like to warmest welcome.
Suaranya kurang besar, maaf, suaranya kecil.
Now, first of all, on this wonderful occasions, I would like to give my warmest welcome to the Honorable Rector of Universitas Brawijaya, Prof. Widodo, SSE, MSc, PhD, Math Science, the Honorable, the Dean of Faculty of Agriculture, Dr. Insinyur Damanhuri MS, the Vice Dean of Academic Affairs, Dr. Sujarwo, SPMP, and the Honorable, the Head of the Plant Pests and Disease Department, Mr. Lukman Kurota Aini, PhD, and the Honorable, the Head of the Agricultural Entomology Department, as well as one of our keynote speakers today, Mr. Ahmad Rizali, SP MSc PhD, and the Honorable, all of the professors and lecturers in the Plant Pest and Disease Department, Faculty of Agriculture, Universitas Brawijaya. And now I would like to give my greatest welcome to the honorable guest today, Mr. Arif Hidayat, the Deputy Chief of Mission Embassy of the Republic of Indonesia in Riyadh, Saudi Arabia. Welcome, Mr. Arif Hidayat. And welcome to the keynote speaker from King Saud University, Saudi Arabia, to the Honorable Prof. Muhammad Ali Al-Saleh, the Honorable Prof. Yasir Eid Ibrahim, the Honorable Prof. Abdul Rahman Saad Al-Dawud. And we are so honored to have all of you here. And thank you so much for your valuable time. And
thank you very much.
Thank you. And welcome to all of the participants. And thank you for being here with us today. Now, I would like to read our agenda today, which are, the first, we will have welcoming speech from the Dean, followed by the opening remarks from the Rector of Universitas Brawijaya. Then we will have opening speech from Mr. Arif Hidayat as the Deputy Chief of Mission Embassy of the Republic of Indonesia in Riyadh, Saudi Arabia. Then we go to the main agenda that will be delivered by Prof. Muhammad Ali Al-Saleh as the first keynote speaker. Then for the second speaker, we will have Prof. Yasir Eid Ibrahim. And the second section will be delivered by Dr. Ahmad Rizali from Universitas Brawijaya and Prof. Abdul Rahman Saad Al-Dawud from King Saud University. And after each sections, there will be Q&A or panel discussions led by the moderators. And at the end, we will deliver the certificate of appreciations and closing remarks from the Dean. Well, to take the advantage of time, let's start this agenda by inviting the Dean of Faculty of Agriculture, Dr. Insinyur Damanhuri MS, to give his welcoming speech. For the Dean, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon, ladies and gentlemen. First of all, let us express our gratitude to the presence of Allah SWT who has given this blessing so that we can organize this international seminar. Next, allow me to express my appreciation and gratitude to all the committee and the Plant, Pests, and Disease Department who have prepared this international seminar. This international seminar on plant protection will of course be beneficial for the development of science, especially in the field of entomology. We welcome Profesor Widodo, SSNI, MSI, PhD Medical Science, Rector of Universitas Brawijaya, and Mr. Arif Hidayat, Deputy Chief of Mission Embassy of the Republic of Indonesia in Riyadh, Saudi Arabia. Also, for all the keynote speakers, Profesor Muhammad Ali Al-Saleh, King Saud University, Saudi Arabia, Profesor Yasir Eid Ibrahim, King Saud University, Saudi Arabia, Profesor Abdul Rahman Saad Al-Dawud, King Saud University, Saudi Arabia, Dr. Ahmad Rizali, Faculty of Agriculture, Universitas Brawijaya. We hope that this international seminar will not only increase knowledge in the field of entomology, but also become the opportunity for international cooperation, especially between Universitas Brawijaya and King Saud University. Finally, I would like to thank for participating in this international seminar organized by the Plant, Pests, and Disease Department with the hope that it will enlighten us, especially those who are always involved in research, learning, and application in the plant, pest, and disease field. Thank you for your attention. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for the opening speech from the Dean of Faculty of Agriculture. And we are sorry that the Rector of Universitas Brawijaya cannot do the opening remarks due to some important business. So let's step on to the following agenda. The opening remarks, I would like to invite Mr. Arif Hidayat as the Deputy Chief of Mission Embassy of the Republic of Indonesia in Riyadh, Saudi Arabia, to give an opening speech for this event. For Mr. Arif Hidayat, the time is yours.
Thank you very much. Very good morning to all of you, and good afternoon for those who are in Indonesia. Allow me to introduce myself once again. That my name is Arif Hidayat. I'm the Deputy Chief of Missions, Embassy of Indonesia in Riyadh, and I'm in the capacity to represent the Indonesian ambassadors who, due to some circumstances, he cannot attend this seminar. First of all, I would like to mention the prominent and resource persons of the seminars today. both from the King Saud University and as well as from the Brawijaya University in Indonesia. There are some persons that I would like to mention here. Professor Yasir Ibrahim from King Saud University, Director Ahmad Rizali, PhD from Brawijaya University, as well as Professor Muhammad Ali Saleh from King Saud, and then Professor Abdul Rahman Al Dawood, also from the King Saud, as well as the Dean and also Professor Widodo, and all colleagues and students of the Brawijaya University. Thank you very much for inviting the Indonesian Embassy to this seminar. Embassy of Indonesia welcome warmly the initiative of cooperation between the two universities, and hope that the result of the seminar will be beneficial for the cooperation of the two countries. I would like to speak like this, that since that I don't have any expertise in the field of agriculture, as well as the sub-item that mentioned or used by the team of the seminar, allow me to speak in general aspect relates to the international cooperations. This is something that I'm dealing with every day. So 3 points that I would like to share with you today. First, I'm talking about the global situation currently now. We are seeing the what you call it, the situation that dominated by the rivalry, by the tensions in some area in global. Impactly, especially for the people, we have seen some impact that really relates to the basic need of the people. I'm talking about the food security. So one thing that I want to say in the first point that here is that I think these seminars relates to the issue of food security, because the agriculture sciences, the end product is the food needed by everybody. So hopefully the seminar will result the contribution of the two countries to tackle the issue of the food security globally. This is number one. Number two, allow me to mention about the relation between the two countries, between Indonesia and Saudi Arabia. I would like to say here that the two countries, even the two people, have had been a long social and cultural interactions. I'm talking about maybe the 2 or the 3 centuries before. Now, mainly the relation or the cooperations dominated by the officials of the two countries. I believe once again that the cooperation on the relation of the two countries can also be done by the people-to-people interactions. And I'm sure that this kind of seminar, this kind of cooperations, especially the U-to-U cooperation between university, can be one of the main pillar, once again, one of the main pillar of the cooperation of the two countries. And I believe, once again, that the education sector can play a very significant role how to develop the two countries into the situation that bring the benefit of the two people. And the last thing that I would like to mention as a third point, allow me to say that I have been here for the 2 years in Saudi Arabia, and I have been witnessing the very massive development in Saudi Arabia. Now, I would like to mention the special mention to the Saudi Vision to 2030. This is very aggressive visions from the leader of the Saudi Arabia, how to make Saudi Arabia become the new hub of the social and economic in the Middle East. This is very good opportunity for Indonesia to utilize this kind of situations. And I do hope that from this seminar can be the contribution from Indonesia, who can play a significant role in achieving the Saudi Vision 2030. Finally, once again, I would like to thanks for the invitations to the Embassy of Indonesia, and wish you all the success of the seminar. And thank you very much once again for the having this kind of initiative. It's really, we welcome this kind of initiative cooperation, and wish you all success. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much for the opening speech, Mr. Arif Hidayat, and thank you so much for your time to be in the international seminar. Now, let me begin with the first sections that will be delivered by Prof. Muhammad Ali Al-Saleh and Prof. Yasir Eid Ibrahim from King Saud University. But first of all, I'd like to introduce our moderator today, Mr. Muhammad Ahid Shibli, SP, MP, PhD, and he will be the moderator for the first sections. Mr. Shibli is the plant pest and disease department lecturer, and his expertise is in plant pathology. Mr. Shibli will help to read the keynote speaker CV and lead the panel discussions in the first sections. And the presentation for each speaker will last around 30 minutes. And for the participants, if you have any questions, you may write your questions in the Zoom chat box, or you may use a raise hand feature to ask the presenters directly. To save the time, I will hand it over to the moderator. Mr. Shibli is already here. So Mr. Shibli, the time is yours.
All right, thank you so much, Ms. Hafida. Can you hear my voice clearly?
It's clear.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Hello, everyone. Good afternoon for Indonesian side and good morning for the speakers. And I'm Muhammad Ahid Shibli. You can call me Shibli, the moderator of today's webinar. A warm welcome to all of you. We are here today to listen and engage with two professors from King Saud University to share ideas how to tackle the plant diseases and get new insight how researchers in Saudi Arabia develop their method to tackle this problem in desert condition. Today is a special day where through this guest lecture program, we are initiate for a collaboration in education and agricultural research between two universities, Universitas Brawijaya and King Saud University. Ladies and gentlemen, allow me to introduce our first speaker, Professor Muhammad Ali Al-Saleh. Hello, Professor Al-Saleh. How are you? Are you already in the room? All right. Allow me to read his curriculum vitae first. So his affiliation in Saudi Arabia is King Saud University. His expertise in virology. He is the head of the plant protection department at the College of Food Sciences and Agriculture, King Saud University, Saudi Arabia. He is also the Deputy Dean of Student Affairs at King Saud University. Professor Al-Saleh is a plant pathologist with expertise in the field of virology. His research interest is concerned with phytopathogenic viruses and the disease they induce. He has conducted molecular and applied research for identification and characterization of plant viruses with the ultimate goal of managing the diseases incited with these agents. He is also interested in the study of other phytopathogens, such as bacteria and virus-like agents. He has published numerous articles in specialized scientific journals in the area of plant viruses and phytopathogenic bacteria. So, ladies and gentlemen, before the presentation begins, let me inform how the presentation will be going on for this first session. So first, Professor Al-Saleh will be invited to present the materials for 30 minutes. After that, the presentation will be continued by Professor Yasir Ibrahim for 30 minutes. Then there will be a questions and answer session for 20 minutes with the audience. So I would like to say hello again to Professor Al-Saleh. Prof, are you already in the room? Hi. Can you hear my voice, Professor Al-Saleh?
Assalamu alaikum.
Wa alaikum salam. Really happy to see you here.
Thank you very much. I am so happy too. Should
We
I
are very-
start?
Yeah, sure. Now, Prof. Al-Saleh, the room is yours. You can share your
Okay, how
material.
about the screen? We are sharing the screen with you.
Yes, please. But your camera is still off.
Okay.
Maybe you can turn on first and we can see.
بسم الله الرحمن الرحيم.
Okay.
الحمد لله رب العالمين والصلاة والسلام على أشرف الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه أجمعين. First of all, I would like to thank the Plant Pests and Disease Department, Faculty of Agriculture and University of Brawijaya in Indonesia. This is really, for us, it is a nice and a good opportunity to share this knowledge together. And also, in the same time, we would like to thank the Excellency of the Indonesian Ambassador in Riyadh, Saudi Arabia, Dr. Abdulaziz Ahmed. And also, we would like to thank Dr. Badr Saleh for his organization, and also our used to be our students, but now he is a doctor, Dr. Arya, for the arrangement of this nice inshallah collaboration and meeting. And inshallah, I'm going in this time to go through different aspects of plant viruses infecting different crops, either in Saudi Arabia or different places in Indonesia. So I am focusing inshallah on these viruses plus the phytoplasma. So should I start? Everything is okay with you? Do you hear me? Do you see the presentation?
Yeah, we can see clearly. Your voice is also clear. Please, you can continue. Yeah.
So this is the title of the presentation, is plant viruses and phytoplasma. When I am talking about the viruses, basically the viruses consist mainly of two things: the capsid protein and plus the RNA or the DNA, which is the nucleic acid. So viruses are it's not like look like fungi or bacteria. We can see it under the microscope. It is we can see it in the electron microscope. It is sub-microscopic nucleoproteins and infectious entity having no specialized system for food and reproduction. Strictly obligate parasite, cannot live outside any body. So it must be in the parasite inside the cell. In nature, it's having a protein, the capsid, which is the over the nucleic acid and the core position for the genome. There is, in fact, for the viruses, either to find either DNA viruses or RNA viruses. There is no virus contained with both nucleic acids. So there is two types based on the nucleic acid viruses. There are DNA viruses, either single-strand DNA or double-strand DNA viruses, and also the same for the RNA, either single-strand or double-strand RNA viruses. Either sometimes is it minus sense or positive sense. So according to International Committee on Taxonomy of Viruses, they call it ICTV, which is well known in the world for the classification of viruses and also other organisms like sorry?
It's all right. Please.
Yeah. So this organization, it has a charge worldwide for the classification, for the identification of these organisms. So according to the ICTV, there is 6 orders, 32 families, and 141 genera, comprising around 1900 viruses species now, or it's already established, and at the same time there is many viruses still under the to be submitted or to be classified in this organization. So when are going to diagnose these viruses, it is important not only to rely only on the symptoms. You have to have many things about this. So the symptoms, almost all the virus diseases seems to cause huge degree of reduction in yield, and the length of the life of the virus infected plants is usually shortened. So as my colleague here talked about the food security and the importance of that, of course, no doubt, these organisms, either viruses or others, they are causing a huge, sometimes reduction for the yield of these crops. So it is very important to manage these diseases, and it is in fact for this reason this seminars or this international seminars is focusing in this area, or this important, in fact, this area. The most obvious symptoms of virus infected plants are usually those appearing on the leaves, but some viruses cause strictly symptoms on the stem, fruits, and roots. So basically, I'm going just to go through the most virus in our lab, which is around more than 30 years. We are just following and collecting and identifying these viruses. So this is just summarizing what we find here in Saudi Arabia. In fact, it is the same, and it is in worldwide in different places, even in Indonesia. But very important things we should focus, or we should have to know. Sometimes the viruses, isolates, and the strains, they are different from place to place, from country to country. Also, the variety of the crops cannot. Some of them can tolerate the virus or the infection of the viruses or the bacteria or whatever, or sometimes other like the environment. Sometimes it's affect the expression sometimes of the symptoms and infect also the severity of the disease. So there is 2 types of symptoms, especially in the leaves. Some of them they call it systemic, and the others they call it local. So what's the systemic symptoms? So these kinds of symptoms can spread in the whole plant, from the infection site through plasmodesmata and the vascular bonds. So the virus, when it's infecting the plant, it can transmit it or can transmit it inside the leaves or other through the plasmodesmata, which is connect between the cells, and also can be spread to the whole plant. And there is different kinds of the symptoms for the systemic symptoms, and we are going to go through these symptoms. And the other kind of the symptoms is the local symptom, which is localized in certain place or certain organs of the plants, like localized to a point of infection, do not move to the parts of the infection. And in fact, this is very, very important for the localization. And sometimes the plants inside itself, in these cells, when they recognize the virus, sometimes they are triggering to each other. So they surround the virus and prevent him to to go through the plasmodesmata. So this is sometimes it's a kind of controlling the virus, and they call it hypersensitive reaction. So the most important things for the symptoms of the viruses is they call it the mosaic. So it is between, it is the green spots and yellow spots and different. This is vary from either severe to mild. So sometimes they call it severe mosaic, sometimes they call it motile, sometimes they call it flick. So the mosaic is characterized by the light green, yellow, or sometimes white areas intermediate between the leaves and in the green lands of the leaves, and sometimes in the fruit. We can recognize this these symptoms of the mosaic in the fruit as well. So it is sometimes which gray areas are intermediate between with the areas of the normal color, and sometimes the fruit. The fruit, it has so different colors. The mottling, it is one grade of the mosaic. So it is sometimes if this discolored patches of the variegated leaves are rounded. So it is like what we saw in this picture. You see some lands it is yellow, sometimes it's green. So this is it is different, by the way, from virus to virus, from the variety of the same plants. So this is, for example, this is for the paper mild motile virus. It should look like these symptoms. There is another symptoms, they call it leaf innation. Sometimes this is abnormal. We saw some growing in the leaves like this innation. It is like the outgrowth of the new leaves or face of the leaves is called leaf innation. Sometimes we saw this in nature, and sometimes it is some insects. Sometimes when they are feeding, they are doing it is look like this symptom. So this is, we saw this with the virus called the cotton leaf care virus, which is transmitted by white fly. When the monocot crops, it is different symptoms, and we saw it is like streaking, like either the wheat or the barley or the sorghum. So we like it is baralla symptoms, and sometimes it is these symptoms. It's so in one leaf. It is like it's not one line. Sometimes it's one line, but it is somehow it is different from line to line. There is light, sometimes like mosaic or streaking, sorry, or sometimes it is just heavy. We can see it clearly. So this is, for instance, with the maize streak virus. There is another symptoms they call vim banding. I am just concerning for the symptoms because very important. The symptoms, we saw it in the field. It is a sign. Sometimes maybe it is infected with the virus, but sometimes we have other either agents or sometimes that nutrition deficiency. Sometimes we have to have an experience for this. We have to take these samples to our lab so we can detect either the virus, so using either the ELISA or PCR or different techniques. So sometimes we cannot rely. It's important. We cannot rely 100% on this plant. It is infected with the virus unless we test it in the lab. So the vim banding, it is more green or yellow color area appeared around the vins. So this is sometimes we saw the vin. It's clear. Sometimes we saw the vins. Sometimes it is green. So they call it vin green banding. Sometimes we call it yellow banding. So it is depend on the symptoms itself. Also, there is clearing. Sometimes you sow. This is what I talked about, the nutrition deficiency. Sometimes when you sow the plants, it seems to be either nitrogen deficiency or others. Sometimes these symptoms, it is not easy to distinguish between those viruses that caused by the virus and those symptoms causing by the nutrition deficiency. So you have to have like some experience for this. And even though you must take these samples and also diagnosis in the lab. So in case of vein clearing, there is veins of the leaves becomes prominent and clear. There is also, they called curling and cupping in the leaves. Sometimes when you sow the plants, it's stunted, the leaves is small, sometimes it's cupping. The cupping, sometimes it is cupping upside down, sometimes it is a downside. So in which margins of the leaf curl upward and downward leaves and petals twisted. So it's mainly with the viruses that are transmitted by whitefly, like the bigoma viruses and other viruses. So it is caused look like these symptoms. And this is, we saw it with the tomato yellow leaf curl virus. Stem pitting, especially with the trees, like either citrus, sometimes when it's infected with the viruses, sometimes we saw the symptoms in the stem. So there is they call it pitting in the stem, and it is observed especially when the bark. When we remove the bark, we saw there is a pitting in the bark. We come to how this virus is transmitted. In fact, this is very important. It's depend on the group itself, the virus itself. Sometimes there is a virus, it can be transmitted mechanically. There is no mean of transmitted only just by mechanical, and there is specificity of that. Sometimes those transmitted by mechanical, it's not transmitted by insects or different insects, and sometimes some viruses that are transmitted by certain insects. For instance, in avid, sometimes there is specificity between the insects itself that transmit the virus and the virus, and it is different. Sometimes the virus can propagate inside the insects, and sometimes it is not. It depends on the virus itself, the group itself. So mainly it's the transmission. It can be achieved either by mechanical transmission, some by vector transmission. There is many vectors that they can transmit the viruses. There is by insects, this like either aphid or whitefly or other insects like leafhoppers, and also can be transmitted through nematodes and fungi too. There's some viruses they can transmit it. Also mice, they can transmit certain viruses. And sometimes there is a relation between the virus and the vector, and sometimes just there is no relation, just only transmitted the virus mechanically. Also, there is another transmission by seeds, by the propagated materials, and also it can be achieved through the parasitic plants like dodder and others. And if there is any time, I can just go through fast about the transmission of the insects. Sometimes there is a relation, either they call it persistent manner, or there is a semi-persistent manner, and also there is no any relation between the virus and the insect. And sometimes the virus is propagated, sometimes it's not. I will go just through some examples of these viruses, especially for different crops. We will focus for the viruses that's infecting the main crops in Saudi Arabia. as well as in other countries worldwide. For instance, in tomato viruses, there is many. There is, of course, there is sometimes 100, but there is many viruses or important viruses. It can be can be infected by this crop, and also it's different from in groups, in families, in the mean of transmission. So, for instance, we will just go for tomato mosaic virus. There is abbreviation, they call it TOMV, and it is belong to viruses and to Bomaviridae, the family, and it can be transmitted mechanically. And for instance, for the tomato spermivirus, it's the abbreviation TAV, and it is belong to Cucumoviruses, Bromoviridae, and can be transmitted mechanically and by avid. And it's look like this. And there is the symptoms, it's vary from virus to virus, from cultivar to cultivar for the tomato, and also the environment sometimes it's play a role of the expression of these symptoms. For instance, this is viruses or this species of these viruses we already detected in Saudi Arabia, and we published papers, and sometimes there's a graduate students, either master or sometimes PhD, they are working on these viruses. For instance, this virus, the tomato black ring virus, we have it in Saudi Arabia, and it causes a problem sometimes in certain area in Saudi Arabia, especially for the growers who's planting the tomato under the greenhouse condition. So this virus, it is belonged to the family Cucurbitaceae that was first discovered in 1946, and it is a member of subgroup B of the genus Niboviruses. And Niboviruses, it can be transmitted through the nematodes, and it is easily transmitted mechanically by the sap, and also infect many plants. And the nematode and the seeds is the main vector of this this virus. The other virus, they call it tomato brown rugose virus, and it is very important virus, and also it is detected in Saudi Arabia, especially in the greenhouses, and it caused a problem with some farmers. And in fact, the important things for those these viruses and how to just to limit their threats to this crop, it is mainly the resistant cultivars. And this virus discovered or described in 2015, and the main host is the tomato and the pepper. And this virus is transmitted by the mechanically. And we saw the fruits, how it look like, and there is some patches, and sometimes the whole fruit, it is not ripe and give the nice color, the red color. Sometimes there is a patches between the green one and the red one. And the other virus, they call it tomato fluorosis virus, and it's the same. It's belong to craniviruses and cholesteroviruses, and that can be transmitted by white fly, and it's already detected here in Saudi Arabia. The other virus, which is very important worldwide and transmitted by the white fly, they call it tomato yellow leaf curl virus, and there is many now species of this virus, and many also there is strains of this virus, and it is transmitted mainly by the white fly. And the white fly, it is very, very important here in Saudi Arabia, and it's not easy to be controlled when it is coming, especially inside the greenhouses or in the open field. And we saw in this picture how it look like the infected plants, how it is stunted, the leaves, it's too small, and sometimes the plants, when it's infected early, sometimes there is no fruit. The other virus, they call it the tomato spotted wilt virus, and this is very important virus. It is transmitted by thrips and also transmitted mechanically. And really, this virus infects maybe more than 500 species plants. So this is very important virus. And also we detect it here in Saudi Arabia. We detect it in tomato and other crops too, like potato. The chili or the pepper viruses, there is many. We detect maybe around 15 or more viruses here in Saudi Arabia in our lab. So there is like a pepper mild mosquito virus and potato virus A, potato virus X, and pepino mosaic virus. All of these infect the pepper in the either in the greenhouses or in the open field in Saudi Arabia. So we saw just some of the symptoms in the greenhouse or in the open field. We saw different type of symptoms for the when it is the plant infected. We saw sometimes mosaic. We saw sometimes, of course, stunting. We saw different type of symptoms. It's depend on the sometimes in the cultivar itself. For instance, for the pepper mild mottle virus, we saw it clearly sometimes in the field or sometimes in the greenhouses, and it showed stunting.
Itu di atas itu nih. Tadi betul lupa yang satunya lah.
It is a stunted plant. It has small leaves, and there is a patches between I mean the mosaic between inside the leaf itself. Sometimes there is they call it the plasters in the leaf, which is sometimes it is coming over. I mean the green one. It is, you saw it, I mean, clearly, and when the crop or the plant it is infected early, sometimes there is no fruit for this. And for the cucurbit viruses like squash, like cucumber, like other cucurbits, there is many viruses we already detected in Saudi Arabia, either in our lab or other labs in other university. And there's many viruses. It look like, especially the zucchini yellow mosaic virus. It is the main virus in infecting cucurbits in Saudi Arabia, and as well as other viruses like watermelon mosaic virus and watermelon chlorotic stunt virus. And the same, those viruses sometimes it is depend on the virus itself or the species of the virus. Sometimes this is transmitted either by avid or by mechanical transmission, and also sometimes by the white fly. We saw just some samples, I mean some symptoms of those. It look like, for instance, for the virus, the zucchini yellow mosaic virus, it is a putty virus, and really this virus, it is cause a major, I mean, destructive disease in the either in the greenhouses or in the open field. It is transmitted by avid in a non-persistent manner, and we saw it is severe. This virus, it is severe, especially when it is infecting either the squash or the cucumber. When it's infecting early, sometimes it's destroyed the whole field. And sometimes when the symptoms or when the isolate the isolate is what's they call it, virulent. Sometimes it's showed, it's like the whole leaf, it is look like the show stream, it is look like the what they call it, the
shoelaces
or shoelaces, yes. So you cannot see even the leaves itself. This virus, the watermelon chlorotic stunt virus, there is one student, he worked in this virus, and really this is very important virus, especially for the watermelon. And this virus is transmitted by hot fly, and it is, you can see it far away from the field. You see the yellowing of the infected plants. So this virus is very important when it is infecting the crop early. I mean, early stage cannot get any yield. And this is very important, and it can infect also other crops and cucurbits like either melon and muskmelon. Other viruses, cucurbit yellow stunting disorder virus. This is very important. Cranny viruses and viruses, and also it can cause a yield reduction, especially when they're affected early, either in the greenhouses or in the open field. And we detect this virus, and there is different isolates of this virus. We saw some of them, it is severe, infecting the crop, and some of them it is just mild, and also sometimes it's give mild symptoms, and sometimes those used to be used for the cross protection to avoid the severe isolates infecting the crop. Other viruses, cucurbit chlorotic yellow virus, especially sometimes we notice when we are visiting the fields, or especially the greenhouses, we saw in the fields, the fields or the leaves, especially the elder leaves, it is show with the symptoms, while the other leaves, especially the new leaves, there is no symptoms for this. And this is main, especially for this virus. I mean, the cucurbit chlorotic virus. Sometimes this is we distinguish this virus, especially for this symptoms in the field. Other crop which is very important for everyone, which is the potato viruses. So there is a virus X, and also the potato mop-top virus and virus Y. And in fact, we have a project, so we detect at least 15 viruses infecting potato. And in fact, that time was the AMV. It is very, very important virus infecting potato in Saudi Arabia, which is sometimes it is unusual in other countries when there is any survey for these diseases in potato. So either BVY or BVX or potato mop-top virus, they are infecting the potato and can give reduction of this crop. Also, as I said, AMV or alpha mosaic virus, even it is infecting alfalfa and clover, but it can be infect the potato, especially when the growers, they are growing alfalfa side by side with the potato. So it is transmitted the virus either mechanically, easy transmitted, sometimes with the farmers or the workers they are working in the field, or sometimes by the aphids when they are moving from this crop to other crop, they are transmitting this virus. For the bean and bee viruses, we detect many viruses like bean yellow mosaic virus, and it is a pathogenic virus in the genus Botivirus and Botiviridae. This virus is transmitted by over 20 species of the aphids and by mechanical inoculation. And also it can give different or the symptoms, it is vary from the crops itself, from the beans, and also from the virus itself. The cow bee mild mottle virus, it's already detected and has different symptoms, can be transmitted by white fly. The same for the bee seed-borne mosaic virus. It is detected in Saudi Arabia in our lab, and it can cause reduction. It is transmitted by the aphid with non-persistent manner for the more than 20 different aphid species. Same for the bean common mosaic virus, which is very important virus in infecting beans. And for the lettuce, there is the virus lettuce mosaic virus, which is very important infecting lettuce. And it is this virus, it's belong to Botiviruses, can be transmitted easily mechanically, and also can be transmitted by the aphid. Also, the onion and garlic viruses, there is many viruses we detected lately in our lab, and there is one student who is working in this viruses. in either an onion on the garlic. So we detect several viruses. It can cause these symptoms, look like either mosaic and abnormal for these leaves. We saw it, I mean, clearly in the field, especially in the early infection. So it can cause a reduction of this crop. For the monocot, I will just go through it fast. There is many viruses we detected in our lab in Saudi Arabia for either for the maize or for the barley and also for the wheat. And this is the list of the viruses we already detected, and the family and the genus and the type of the mean of the transmission of these viruses. By the way, I can send this presentation or this seminar or this slides to anyone he would like to share it with us. This is, for instance, for the mosaic and soil-borne wheat mosaic virus. We saw these symptoms in the field, and we saw it mainly for either for the wheat and also for the barley. We saw the same symptoms because this is a monocot viruses or plants. Sorry, this is for the barley yellow dwarf virus. We saw the symptoms, and the same for the barley yellow mosaic virus. We saw that, and we detected in our lab. For this Johnson grass mosaic virus also detected, and the same for the maize chlorotic mottle virus and the sugarcane mosaic virus. The same, we saw these symptoms. It is this virus sharing the same symptoms. Same for the maize, barley, and also for the wheat streak mosaic virus and the wheat dwarf virus. It's already detected and sharing somehow the same symptoms. For the fig viruses, which is very important in Saudi Arabia and also in Indonesia and other country, we saw or we detect many viruses plus the viroids also. So this is the list of the viruses and the viroids already we detected in our lab, infecting the fig crop. And this is the symptoms we noticed in the field. In the left, there is a asymptomatic leaf of the fig, and also this is the different symptoms we noticed depend on the virus and depend on the cultivar itself. For the grape viruses, there is around 10 viruses we already detected, and it is belong to different genes. Some of them, one of them, it is not signed yet in the family, in the ICPV, and some of them it is transmitted mechanically. Some of them it's not transmitted, just transmitted by especially the Marybug. And we notice these symptoms in the field. Sometimes it is light mosaic. Some of them just light, or they call it flick when it is just only small dots or yellow dots in the leaf. Also for the citrus viruses, we detect some viruses like citrus tristeza virus, which is very important virus infecting the citrus, either for the lemon or for the grape. Other viruses, or this is the main. There is other viruses we already detected, especially for infecting in the nurseries, and also for the infecting the weeds outside the field, which is very important. Other type of this seminar, we will focus just for the phytoplasma. The phytoplasma is characterized. The symptoms is abnormal floral organs developed, including phyllody, which is very important. The symptoms where it is noticed in the phytoplasma when it is infecting the plants, and we detect some of these, or maybe 5 or 6 infecting the crops. Look like the tomato. We saw these symptoms. It's clear for when it is infecting this crop. So there is, especially it is look like look like this the plant, it is just a bushy. You cannot, there is no, cannot give more either fruit nor also the flowers. And this is the same, the typical symptoms we saw in the field when it is infected, this crop with the tomato with the phytoplasma. The same for the pepper. It is when it is infected. This, by the way, the phytoplasma, it can be transmitted by transmitted by insects like the leafhopper. We saw it in the field. We collect these the insects. We already detect even the virus in these insects. The same for the eggplant, and also
Professor Saleh.
the alpha.
Professor Saleh, Al-Saleh, Professor.
Yes.
You have 5 minutes left for this presentation.
Yeah, I will finish inshallah before.
All right. Thank you.
So the alpha the same. You will see this is in the field. So small, the plants is bushy, and you cannot see any flowers when it is infecting by this phytoplasma. And also for the faba bean, the same symptoms. And also for the mango, which is, this is strange. Sometimes it is rare, but we find it infecting the phytoplasma, infecting the mango. And it's the same, the symptoms. You will saw small leaves, and it is bushy. And when we get it, and we get the samples, we detect the phytoplasma from that. And this is with different crops, the same. And also, it is very important in Saudi Arabia, the palm, the date palm, it's very important. And there was a project in the eastern part of Saudi Arabia for there is an important disease, they call it arujam. So they, after maybe 5 years of investigation, because the palm, it is not easy to work with. So they found there is a phytoplasma infecting this and causing this disease in this crop in Saudi Arabia. So why we are studying those viruses and this phytoplasma and the bacteria and the fungi and others? What's the main? It is must to know what's the kind of the organism and the kind of the virus or the kind of the growth. It is important to manage these diseases. So management, it is start first with the right or the diagnosis of these viruses or these organisms. Then what's the important? Unfortunate, the viruses that there is no either chemicals or antibiotics. For instance, we can treat the plants after infection. While the virus, when it is infect the plants, that's it. There is no cure for this. So mainly this is it is look like the name of our department, plant protection. We have to protect the plant. So it is just prevention to prevent the virus to come to the plant. So we can just say, for instance, avoiding exporting or importing of viral disease plant material to disease pre-localization through quarantine, law certification and inspections. This is very important, especially the moving of the materials or the seeds from country to country, even from region to region. This is very important. You have to have those certified. It's not there is virus free. Also the selection of virus disease free seeds from the disease free regions. Sometimes we find in the seeds it's written on the label, this is for instance free of viruses, or sometimes there are resistance of this virus and this virus or this virus. For instance, this coming from either Europe or from America, maybe the varieties of these or isolates of these viruses maybe cannot infect. But when it's coming in, for instance, in Saudi Arabia or different country, maybe there is other other isolates, and it can infect this crop or this this crop. So it is very important when we are just bringing new seeds from different companies. We should inspect those seeds and make sure it is for sure it is not or virus free, and also as written in the label, it is either resistant or tolerate. So also the selection of virus disease-free planting materials like cutting or like rhizomes or tubers is very important to be tested and make sure there is no virus. Application of soil fumigations between foreign nematodes that transmit the virus is very important to be done. So destruction of weeds that serve as a host of virus causing virus or viral disease in plants is very important, especially around the greenhouses and also the fields, because if the main crop is not there, the insects they are carrying this virus, it will go to these weeds. So the weeds must be destructed. Also, the cultifying the resistant varieties is very important, because this is the main important of controlling plant viruses. So the resistant cultivar is very important. Also, the application of temperature treatment, sometimes it can just relieve or can take, especially for the materials or plant materials, it can take the virus, especially because it is sensitive to the temperature. Also, application of insecticides will control the insect vectors that serve as a host for virus causing viral disease in plants, which is very important. Also, especially for the guys who they have the greenhouses, it's very important using the screen. They cannot allow the virus to enter inside the greenhouse. And also the double doors is very important. So you cannot avoid, you can avoid entrance of these insects, especially when they are carrying the virus. So this is just only it's not I mean a picture of these viruses that we study in Saudi Arabia, and we share it with you with the main the management of that. And I wish I just give something an important, and we share some information. Maybe we remind some people for this. I know some of them they are experts. Sometimes some of them it's not virologists. So at least we share it, and we hopefully, inshallah, we get benefit from it and from our brothers and in Indonesia. Thank you very much for this time, and appreciate the time you are just waiting and listening to me. Thank you very much. Assalamu alaikum wa rahmatullahi wa barakatuh. You hear me?
Yeah, I hear it very clear. Thank you so much, Professor Al-Saleh, for giving us such informative and interesting presentation. So now we come to the next speakers. Professor Al-Saleh, is that all right?
Yeah, Dr. Yasir.
Okay. So, ladies and gentlemen, now we come to the next speakers with Professor Yasir Eid Ibrahim. He is from the same institution with Professor Al-Saleh, King Saud University, Saudi Arabia, with expertise in microbiology, especially in bacteria and fungi. He is a full-time lecturer at the Plant Protection Department, College of Food Sciences and Agriculture, King Saud University. He is a plant pathologist with research interests is mainly on plant pathogenic bacteria. He also has conducted various works on bacterial diseases of different crops, including identification, detection, characterization, management, and awareness campaign. Aside from that, from Professor Yasir Ibrahim has also conducted research on plant pathogenic fungi. Professor Yasir Ibrahim, how are you? Are you already in the room?
With just a few minutes because I have a problem in my presentation.
Yes, sure.
Yes.
So everyone, if you have the questions, please put your question in the chat box, or you can
Okay, I'm
address.
ready. I am Muhammad Al-Sah. I am ready if there is any question or just if there is anybody he would like to add any information and benefit us. Unless we will wait until Dr. Yasir, he will be ready, inshallah.
All right. So actually, there are some questions from the audience. Professor Saleh from Mr. Pujo. He works in a seed company. He has a question for striking cause of virus and fungi. Is there any difference for early symptom on the leaf?
There is difference between the streaking, you mean?
Yeah, for striking cause because of this virus and fungi, is there any difference for early symptom on the leaf?
As I mentioned, we cannot rely on the symptoms, but the exhibits sometimes when they are coming in many times and we saw these symptoms, we say, yes, this is a virus, but we must go to take this sample to our lab and confirm these symptoms. It is caused by a virus. There is the nutrition deficiency. Sometimes it's especially for the monocot, especially for the wheat, for the barley, for the sorghum, for the corn. Sometimes there is misleading of these symptoms. So you cannot say this is a virus, or you cannot say this is a nutrition deficiency until you confirm it. Either confirm it, for instance, for if it's virus in the detection in the lab, and also it is nutrient deficiency. You have to get a nutrient for the crop, so it will go. So this is mainly the difference. And we are teaching the students you cannot rely for the symptoms to detect the viruses. Yes, it is part of it, but it is not the main. Sometimes the virus is different from. It is sharing symptoms with other things. Even sometimes with aging. Sometimes when the insects they are feeding, they have some toxins, and it can give, especially for the aphids when they are feeding, it is it can like the cup shape, and it is sharing with the same symptoms of viruses. So it is very important for us to diagnose these the symptoms and make sure this is caused by viruses. So I hope hopefully I get some or I answer some of this question.
Yeah, interesting, Professor. So we have some question more. Maybe Mr. Faiz want to propose directly to Professor Al-Saleh. Faiz, are you there? Okay, I will read it for you. So there is question again from Faiz regarding plant virus vectors. Is there any way to suppress the viruses carried by the vectors? For example, the case of using Wolbachia, which can suppress dengue virus. Thank you. Can you follow the question, Professor Al-Saleh?
I don't know how to say. Really, there is. I mean, especially for the viruses that are transmitted by the vector. For the controlling, it is very important to control the vector itself before it is coming to the crop. Sometimes if the vector it is not carrying the virus, sometimes it is easy to be controlled. But when the vector it is carrying the virus and when it is feed, especially feed on the crop and transmit the virus, even if you control it, it is not, it is too late. You will control the vector, but you cannot control the virus. Are there any either chemicals or whatever can suppress the transmission? For my answer, I didn't know, but it is controlling the vector, yes, but about controlling, the transmission, I don't know, because sometimes it is depend on the virus itself, depend on the vector, depend on the relation. Sometimes the relation, it is different. If it is especially for the propagated, there is viruses, it's propagated inside the body of the insect. So this is a problem with this. If there is any way to, using the molecular techniques to avoid this, to block this, yes. There's any genes to be blocked, yes. I'm sure it is at least block the transmission of the virus to the plant.
All right, interesting. So we have question from the questioner again from Mr. Agus Muharram. Mr. Agus, please.
Yes, thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Very short question for Professor Ali. Do you still use the indicator plant for virus strain identification and also for multiplication of the virus for your research? Or are only using the RNA or DNA analysis for differentiating the strain of the virus? Thank you.
Very nice question. Thank you very much. Yes, we are using, they call it biology, serology, and molecular biology for the detection. It is very important to have a propagated plant so we can maintain the virus on it. So we have to have this indicator plants. We have to have, when we are studying or detecting new virus, we have to have many things. First, the indicator plants, of course, either from the literature, we know that the host range of this virus is very important to study. Other, to detect it in serology, like in ELISA, if there is any antiserum for this virus. And also for the molecular biology using the PCR, depend on the genome of the virus, whether it is it is DNA virus or RNA virus. So to have primers to detect it, to sequence it, the whole story to be done, and also to compare it with other viruses. Either the same species in the gene bank, in CPI, or is it a new virus, a new species? We find a new species here in Saudi Arabia. There is one infecting pepper, and all it is the same. Other virus. It is the same. It looks like other viruses. It's already named in other countries. So this is the main important about the detection and the description of this virus. We use biology, serology, molecular biology. Thank you.
Thank you very much.
Ready?
So are you ready, Professor Ibrahim?
Yeah, he's ready now. Yeah.
Okay, nice. So you can share and start now. The room is yours.
Assalamu alaikum wa rahmatullahi wa barakatuh.
Wa alaikum salam wa rahmatullahi.
First of all, I would like to thank His Excellency Dr. Badr Saleh in the Embassy of Indonesia in the Kingdom of Saudi Arabia for his inviting me to participate in this meeting, and also for the committee of the meeting. My name is Yasser Ibrahim. I work as a professor in the Plant Protection Department, Faculty of Food and Agriculture Science, King Saud University. I work in the field of plant diseases, a specialist in the field of bacterial plant diseases. I am interested in diagnosis and identifying plant pathogenic bacteria on fruit and vegetable crops as well as studying the epidemiology of diseases and the methods for controlling them. I have a lot of publication in the plant diseases and also especially in bacterial plant disease. Today, I will talk about the bacteria as plant pathogens in Saudi Arabia. Uh, 30 years ago, when I started working as a graduate student, I liked the bacteria. But after my marriage, I like it more. Why? Because bacteria are cute, in my opinion. Bacteria are singular cell microorganisms, generally ranging from 1 to 2 micron in size. Individual bacterial cell cannot be seen without the use of microscope. However, large population of bacteria become visible as a biofilm on plants, as suspension, also blocking plant vessels or colonies in petri dish. Bacteria are among the microbes that successfully colonize plants as they mature. For beneficial purpose or a pathogen population of 10 to the power of 6 or higher are normally required for bacteria to function as biological control or cause function disease. Although I said the bacteria are cute, plant pathogenic bacteria also cause many serious diseases in plants throughout the world, including Saudi Arabia. Today, I will try to summarize it, which kind of the bacteria in Saudi Arabia. Especially, I will talk about the top 5 plant pathogenic bacteria in Saudi Arabia. First disease is greening or hang long pink. HLB have a different synonym in different countries, like yellow dragon in China, mottle leaf in the Philippines, dieback in India, yellow shout in South Africa, also in Indonesia, phylum necrosis and vein phylum degeneration in Indonesia, look pain or dropping disease in Taiwan. HLB originally thought to be caused by a virus, but now it's known as to be caused by unculturable phylum limited bacteria, Candida staphylococcus. There are 3 forms of HLB that have been described. The African form produces symptoms only under cool conditions and it's transmitted by the African citrus cilidy, Toriosa erythrea. The Asian form prefer warmer condition and it's transmitted by the Asian citrus cilidy, Difronia citri. Recently, a 3rd American form transmitted by the Asian citrus cilidy was discovered in Brazil. In addition, Libribacter pathogens are graft transmission and have been spread through the transport infected vegetable propagated plant materials. The HLB bacteria can infect most citrus cultivars, species, and hybrids, and even some citrus relatives. There is an old saying among plant virologists: when greening comes in, citrus goes out. More than ever, this seems to be true today. HLB reduces citrus tree production by about 50%, and in advanced stages of infection may cause the death of the trees. For example, in Florida, the disease was reported in 2005, and now it's resulted in a loss about 100,000 acres of citrus and an estimated $3.6 billion in lost revenue. HLB and the factors worldwide distributed in more than 50 countries. Based on characteristic symptomology, HLB was initially reported in Saudi Arabia during the mid of 1970. However, the presence of candidates and the Asian citrus psyllid vector was not confirmed until 1981, when they were detected by electron microscope. Our objective in the last 5 years, we made a survey in all Saudi Arabia citrus producing region, and our results showed the detection of the bacteria and confirmed only the identity of Candidatus Libribacter asiaticus collected from citrus leaf and fruits in 10 different regions out of 13 in Saudi Arabia. This map shows the region of Saudi Arabia where the disease and the vectors were reported. For the molecular characterization based on the 16S rDNA partial sequence, all Saudi Arabian isolates collected are related to the Candidatus Libribacter asiaticus. And to understand the genetic variability, demography, and invasion history of the Asian citrus psyllidae and the Candidatus phasosystem in Saudi Arabia, we're working in 3 markers for Asian citrus psyllidae. This is the mitochondria gene and the chromosomal gene and the endosymbiotic Wolbachia gene. And for Candidatus, we worked in 16S Candidatus associated full length for the prophage genome. And the data showed for the psyllidae, all psyllidae collected from Saudi Arabia related to one type only, and also related to Iran, as shown in the phylogenetic trees. Based on the population of the nucleolic gene, also we have a lot of variation between psyllidae based on the regions in Saudi Arabia. Based on the Wolbachia surface protein gene, also we have some variation between isolates in Saudi Arabia collected from different regions. For the prophage, we have 2 types reported in Saudi Arabia, type 1 and type 2. The first type related to the Pakistan isolates, and the second type also related to Pakistan and also Florida. As all know, especially working in plant diseases, there are no practical cure for HLB disease. But most of the work just to how to solve the problem as we can. And also we try to make some chemicals as inducer resistant, like salicylic acid and phenyl acetic acid, in combination with some rootstock, different rootstock cultivated or used in Saudi Arabia. We try to make something about to help the farmers to use something to solve the problem, especially in the first stage of the infection. And we get some promising data with salicylic acid and also phenyl acetic acid in combination with antibiotic or without antibiotic with different rootstock cultivars. We try to follow the relative expression of pathogenesis-related gene inside the plant after the infection and after the treatment with the chemicals also. We measure this pathogenesis-related gene. And alhamdulillah, after that we have 4 publications in greening in our lab. And this is the part of the PhD student Arya Waidawan from Indonesia. And he worked maybe 10 years ago with us in the master degree and PhD degree. And alhamdulillah, he finished his PhD. The second disease also, it's important disease related to citrus is citrus canker. And I know from area and from literature also, both diseases, even citrus canker or citrus greening, make a problem in Indonesia. And inshallah, we hope a lot of collaboration together in Saudi Arabia and Indonesia or other agency to get some solve of the problem, inshallah. Citrus canker, infected fruits, foliage and stem. The symptoms is spongy erupted at first, corky rough lesion with raised water, soaked appearance later in the middle of lesion. Citrus canker in Saudi Arabia also reported in 1981 as a disease, not like symptoms, but no publication was done in Saudi Arabia from 1980 until 2010. In 2010, we have typical citrus canker symptoms were observed in some south region in Saudi Arabia, namely Al-Baha, Asir, and Jazan. And in same farm, interestingly, other citrus susceptible trees, like lemon trees, near the infected one, didn't show any symptoms. And this is more than one pathotype maybe exist in Saudi Arabia. And due to this observation, we try to isolate the bacteria and make some physiological biochemical tests and also DNA sequencing. And based on our data and pathogenicity test, we have two different types of symptoms were produced by the Xanthomonas strains. And at that time, we identified we have strain A. This is the Asiatic citrus canker, and this is the common strain worldwide. And another one is A star, and this is only in Asian countries and reported in 1998, and also present in Saudi Arabia. Using some BCRs techniques, we try to find the variation between some isolates, and we find some, but it's not 100% sure. Using the primers or old conventional primers to differentiate between the strains. And for this reason, we make some collaboration with some institute in French, for example, Saud, and we send a lot of collection from isolates to analysis using the recent techniques called the multi-locus variable number of tandem repetitive analysis, or MLFA. And this is a high technique used to differentiate between strains from XCC. And the data showed Saudi Arabia sure have two strains A and A stars, and also we identify subclade under strains. Also, Saudi Arabia have subclade 4.1 and 4.2. We try to more variation between some isolates, and also we use the CRISPR genotyping for a lot of Xanthomonas in our lab. And we finding Saudi Arabia only hosted 13 spoilotype out of 63 spoilotype recorded worldwide. And this is too much spoilotype reported in Saudi Arabia, and this is means a lot of introductions come to Saudi Arabia and a lot of strains. To find the possible disease management, we try to use the copper-based bactericides, and it's routinely applied to control Asiatic citrus canker in 3 Saudi Arabia grown regions. However, the extensive use of copper can lead to soil contamination, and also it has direct harmful effect on both human and the surrounding ecosystem. In addition, excessive use of exposure to copper can select XCC strains that are copper resistant. Until now, copper resistant XCC strains have not been isolated from Saudi Arabia, but they could appear with the continued reliance upon copper bactericides for ECC management. Also, we try to use the systemic acquired resistance in the users, and we get some promising data showing activity against ACC when applied in combination with copper with Pseudomonas fluorescens or Bacillus subtilis. Also, we try to use the bacteriophage, another promising alternative strategy, and we get some promising data under field condition or greenhouse condition. And this is our publication related to citrus canker, related to our lab. And this is the recently publication in 2022 in the Evaluationary Application Journal. And also, alhamdulillah, we have accepted in Plant Pathology Journal for the last paper about the characterization of XCC using the CRISPR typing. We have a lot of publication in cancer, also including area, because also his master degree also in citrus canker. And also when we come again for the PhD, we start working again in citrus canker. The third disease has a economic input also in Saudi Arabia. This is the bacterial spot of tomato and pepper. The pathogens are Xanthomonas eophysicatoria, Xanthomonas perforans, Xanthomonas physicatoria, and Xanthomonas gardneri. And the hostess is pepper and tomato. This is some symptoms we collected from different open field and greenhouses related to tomato and pepper. And this is a map show the geographic distribution of different species of bacterial spot of Xanthomonas worldwide. And also we have a plan, still we working in the disease to determine which species in Saudi Arabia, present in Saudi Arabia. And also we have still some experiment about the management in the disease under greenhouse or open field conditions. And this is our publication also for all the publication in 2012 and 2011 related to only Xanthomonas physicatoria. But now we also get another species from Xanthomonas, but not published yet. The fourth disease also present in Saudi Arabia, this is the fire blight. Fire blight is caused by the bacterium Erwinia amylophora. It's a gram-negative and short-rod shaped bacterium in Erwiniaceae family. The most economically important hosts are apple, pear, apricot, berries, and mainly in Saudi Arabia, it will be infected the pear. We are not isolated from apple until now, or other host, apricot. Only we isolated from pear. Symptoms like fire blight disease was first observed in 1981 when the apple and pear started to cultivate it in Saudi Arabia, and also in 2013. But the disease status at that time was based solely on visual observation. The claim of the existence of fire blight in Saudi Arabia caused by Erwinia will be investigated in this study. We have a biolog machine also to identify the bacteria, and we start already, and we get more than 20 isolates and confirm it by conventional PCR and also screening for the CRISPR locus. We did some pathogenicity tests in beers also. Also, we test some isolates for copper and streptomycin resistance. Still, the work continue for fire blight, and we have a master degree student from Pakistan. And inshallah, maybe next year we will be finished his thesis. And the last disease also make a problem is Ralstonia solanacearum. And this is gram-negative bacteria, cause bacterial wilt and root of potato, formerly known as Pseudomonas solanacearum. It's a complex species disease, has 5 races and also 5 biofars. The races is determined by the host range of the group. However, biofar is determined by the type of food source they use. The dominant or the isolate bacteria we find is race 3 biofar 2. And why is Ralstonia race 3 biofar 2 important? Because it's in some areas like race 1, it's endemic in the US. Race 3 is the pathogenic on potato and tomato, also worldwide. It's a potential to spread widely by seeds or vegetative material. Bacteria can survive in temperate climates. Also, the susceptible crop for Ralstonia is the eggplant, potato, and tomato, and also pelargonium. The damage caused by the bacteria is the potato only major concern, and based on the literature, the worldwide losses around $950 million, and it's a quarantine disease also in a lot of countries, especially in Europe. And the losses from the destruction of infected plant in USA, too much losses. And we have some publication in 2020 and also in 2012 about the publication in induction of disease defense against Ralstonia. And this is the agencies collaborating with us in our lab, King Abdulaziz City for Science and Technology, and the National Plan for Science, Technology and Innovation related to King Saud University, Ministry of Environment, Water and Agriculture, the Deanship of Scientific Research, King Saud University, the French Agriculture Research Center for International Development, CIRAD. Also, we have some collaboration with the School of Plant Science, University of Arizona, and also Institute of Biological Chemistry, Washington State University. Finally, I would like to thank all for listening and inviting me to participate in the meeting or conference. I will come cooperation with those interested in the field of plant diseases, especially bacterial plant diseases. And terima kasih.
Thank you so much. Terima kasih, Professor Yazir.
Yes, thank you very much.
Actually, I want to ask something to you before the audience, especially because you mentioned that bacteria is cute and more cute after you marry. What happened? What happened, brother?
It's after that are not cute.
Maybe there is a scientific reason for that.
Yeah. maybe.
All right. It's all right. So, ladies and gentlemen, the next is a questions and answer session again.
Okay.
Every one of you, you can put your question in a chat
For
box.
sure.
Yeah,
For sure.
and I will try to read it for you.
Okay.
But if the audience want to propose directly, please feel free to raise hand by
Okay.
the how can I say, the feature in this Zoom, right? So
Okay.
there is a question from Nurfianto. Sorry, yes, here. The first question is how the development biotechnology and plant breeding to make citrus resistant with HLB and the factor Diaporina citri in Saudi Arabia. The second is how is the plant defense mechanism from HLB and Diaporina citri to understand better for the integrated pest management specialty. Thank you. What do you think?
Yeah, for first question, and we have we use different molecular markers to identify the salinity, also morphologically in our department, and after that molecular biology, we have 3 markers to identify, and only we find until now this is only Asian citrus salinity in Saudi Arabia. For the second question.
The second one? How is the plant defense mechanism from HLB and Diaporina citri to understand better if we try to relate this mechanism with integrated pest management and also disease management?
Yeah, for sure, based on what we have, this is for control all. Yani I think all BASO system same for the controlling, but we try to find what we have in Saudi Arabia to see what we can do. Yani based on, for example, to the quarantine. Now we know most of the trees come from outside with visual, and we measure the distance between different regions in Saudi Arabia. There is no much distribution between insects, and this means the insects is not responsible only for the distribution of the disease, but the plant material is distributed. And for this reason, our recommendation for the Ministry of Agriculture about the interior quarantine to prohibit the transport, the seedling from area to another area, for example.
Yeah, interesting. Because without quarantine, we will not able to stop the transmission and distribution,
Yes,
especially.
Yeah.
Yeah. So the next questioner is from Pak Mintarto. Mungkin Pak Mintarto ada di sini. Pak Mintarto want to propose therapy, or you want me to read your question?
How is the mood? I couldn't read again.
Yes. Let me check first. This question is for Professor Ali Saleh. So this is from Pak Mintarto. So Pak Mintarto is associate professor from our department, and he asked about, I want to get information, how is the implementation of integrated control management for virus diseases in Saudi Arabia? Is there any related information about it? And what are the opinion of people and the government of Saudi Arabia about GMO technology for plant virus management? Maybe Professor Saleh want to answer this?
Assalamu alaikum. Do you hear me?
Yeah, very clear.
Okay. So about the IPM, about the controlling of plant viruses. In fact, this is very nice questions. I mentioned before, since the virus is already in the plants, there is no cure, even in the human too. And the COVID-19 is a very, life examples for us when it's blocking the whole world. So the problem with the virus, we have to prevent it. This is the main important for the IPM. We must prevent the virus to come to get in the plant. How the prevention? There is a different ways of that, or different means of that, especially controlling the vector. Other things that is also protect our country through the quarantine, preventing any materials or invested materials with viruses or other organisms to prevent it to come to the area. This is very important. Also inside the farm, it's very important to move from the worker from either from greenhouse to greenhouse. Should be there is a changing in the clothes and the gloves and everything, because sometimes there is some viruses or there is no or asymptomatic symptoms in the plant. So it is must be everything should be considered. It is not only just controlling the vector, or just only quarantine, or also the mean of the the workers. It should be everything. We should consider it, that especially it's mainly depending on the crop itself, and also depending on the when we know the crop, for instance, it's vegetables, we know the viruses that are infecting, we know the vectors, so we must prevent that to come to the plant. It's very important. For instance, for the greenhouses, must be every consideration should be taken, preventing the insects to get inside the greenhouse, either using the screens, using the double door, using the main important is using the cultivars. There is many cultivars for the plant or for the crop. It is some of them it is more resistant, some of them is immune, some of them it is tolerant, most is susceptible. So we consider for the IPM, we consider everything. So we consider controlling, meaning we consider the also removing the weeds and everything. So this, it's not easy answer, and it's not easy to be applied. And it is very important for the farmer to understand everything about his crop and about the pathogens that infect the crop. So this is, hopefully it is answer the first question, the IPM. The second question is about I forget. Excuse me.
Yeah, I will read it again.
The second question.
Yeah. The second question is, what are the opinion of people and the government of Saudi Arabia about GMO technology for plant virus management?
This is a political question, but anyhow, there is no problem. I mean, sometimes in the content of certain, I mean, the cans or something, there is a symbol known whether it is GMOs or not. But anyhow, we do not see any problem with this, especially in our country. So sometimes we are using for certain, but it is under experiment only, using some mediated resistance in the plant to control the viruses. And sometimes we use in the Mbigomo viruses for the TULCV. Also, we embedded the certain elements in the plant, so it will be controlling or preventing the virus to come in. So it is already we accept it under experiment, and which is very, I mean, acceptable. But I mean, in commercially, there is nothing. I mean, we as I know there is nothing. I mean, now we have only just the regular one. Clear?
Or yeah, I think clear. It's similar with our country here in Indonesia, because it's GMO anyway. So many aspects we need to consider again if we need to commercialize the product, right? maybe it seems with the Saudi Arabia government. So the next questioner from Mbak Rasiska, silakan Mbak Rasiska. If you want to propose Darwin, please.
Thank you, moderator.
Yes.
I want to ask to Mr. Prof. Ali Saleh. Thank you for Mr. Ali Saleh for your presentation about virus disease of plant for chili, tomato plants, and each other. In Indonesian, have two sessions. It is the rain session and dry session. In the dry session, we know the percentage and intensity virus disease is very high. Maybe the same in the Saudi Arabia, because the Saudi Arabia have the climate constant high temperature, and I think that the high temperature have effect to the percentage and intensity disease virus. So I want to ask for you, how does government or the scientists or to solve a problem about the high temperature in Saudi Arabia, because the Saudi Arabia have the high temperature the constant in the years. So this is one question for Prof. Ali Saleh.
Thank you for a nice question. But this is a joke first. I mean, we are plant pathologists. We need to see either the viruses or the fungi and the bacteria. So if they control it, and there is no virus, no bacteria, no fungi, where are we gonna work? What are we gonna do? Nothing. So this is just a joke. Now we found and we're study to manage those diseases. In fact, as you said, yes, we are especially in the hot temperature, especially in the summer. We have problem first with the insects, especially with the white fly, with the aphid. So this is problem. Yes, it is in the winter. It's not less important, but during the summer, yes. But unfortunate, whatever we try, I mean, those either insects or the carrying the virus, it is there. And we have really a real problem with especially when inside the greenhouses, because this is a controlled environment, and sometimes when it is coming to the controlled environment, these viruses, it will destroy. So we are trying just the problem. The second problem, we have the problem with the resistance of these insects, especially with the white fly, with the insecticides. So unfortunately, in different areas, especially in the central part of Saudi Arabia and other parts, we have problem with the white fly. Because it is whatever you control it, you spread whatever insecticides, different classes of insecticides. The white fly, it is getting resistance of these chemicals. So we need to have first the prevention of this entering this insects to either the greenhouse, but in the open field, unfortunately, you find a lot of insects there. So really, it is not easy task. It's not easy to manage, but at least you have to have prevention for these viruses of these insects to reach the plant. So whatever we did, we go back to the IPM. We must have the resistant variety to those either to the insect, to the vector, as well as to the virus. Second, prevention. We prevent the insects that carrying the virus to get inside, especially the greenhouses. And really, there is a challenge. It's not easy task, but we are doing we will do our best to manage this and to heal whatever the farmers there. Sometimes they are suffering of those. Sometimes we went to greenhouses, especially for the plastic that's not well controlled. It is destroying the whole. Maybe the farmer, he get a lot of losses, yield losses. Sometimes he gets zero of yield. So this is the life, and this is the how to live with these organisms. Thank you very much.
Thank you. Thank you, Professor. Your information for me. Thank you.
All right, thank you so much, Mbak Raziska, for your interesting question. And I think because we have a limited time of Yasir, of Al-Saleh, so I think we finally come to the end of this seminar. And before I close the seminar, I'd like to thank to the speakers for the very interesting presentation and all the participants for the very active participation. After this, we will have a little and special gift for the speakers from the head of Plant Protection Department, Dr. Lokman Porota Aini. And maybe the admin can help me to share the special gift from our department. Here is the certificate from our department for Professor Yasir and Professor Al-Saleh. We are very happy for having you here as a guest, and hopefully we can meet in person in Indonesia or Saudi Arabia and can make a further collaboration, not only with the guest lecture, but also with research. By the way, if you want to say something to the audience, please, Professor Yasir or Professor Al-Saleh.
Yes, if you allow me, I appreciate that.
Right.
Yeah, really, I am so happy, as well as my colleague, Dr. Yasir and Dr. Dawood, and also the whole of our department, as well as our college. We are so happy to share this information. We are so happy to have this collaboration with our college and your college. And in fact, we have a good reputation for the students, for the Indonesian students. We have many students who graduated here, I mean, or study here in our college, either in College of Agriculture or other colleges, engineering or other. We have, mashallah, good students. They have a good society. I used to have to be in charge for the students' affairs in the university. There is, mashallah, tabarakallah, there is a club for the students, Indonesian students. I joined them several times. Mashallah, tabarakallah, it's good students. They give a good reputation for either for Muslims and for the good scientists. They present Indonesia in a good manner. And we have some examples here. I mean, we have Arya, mashallah, tabarakallah. He is, mashallah, on top of our students. There is another student, Alan. He worked with Dr. Dawood and Sukerno. They, mashallah, tabarakallah, they publish a lot of papers, scientific papers in well-known or famous journals. And we proud of those. Of course, we would like to join, to collaborate, either to come to Indonesia to visit our brothers, our students there who's now scientists in universities or in the centers. And also at the same time, we would like you to come to visit us in Riyadh, sure, and also in Mecca, and make Umrah and Hajj, inshallah.
Inshallah.
Jazakallah.
Yeah, thank you. Thank you so much, Professor. So we are very happy to say we're very welcome. Yeah, actually, Indonesian community will be happy to make food together and over with many friends and make some organize some events and share the food together. Yeah, so interesting. Thank you so much. So everyone, I think it's the end of this first session for this seminar. So please give the applause for the speakers. You can put the speaker applause in the Zoom. So thank you, Professor. Back to Ms. Afida.
All right. Thank you very much, Mr. Sibli. And thank you very much for Prof. Muhammad Ali Al-Saleh for the great materials about plant viruses and phytoplasma, and Prof. Yasser Id Ibrahim about bacteria as plant pathology. Thank you very much for your valuable knowledge and your valuable time. And thank you for Mr. Sibli for leading the discussions. Now, let's move to the second sections that will be delivered by Dr. Ahmad Rizali from Universitas Brawijaya and Prof. Abdul Rahman Saad Al Dawood from King Saud University. And the moderator for the second section is Dr. Sylvie Ikawati. Dr. Sylvie is the lecturer in the Plant Pests and Disease Department, and her expertise is in toxicology of pesticides. So I hope everyone could listen for the explanation thoroughly. And for the participants, if you have any questions, feel free to write down your questions in the Zoom chat box, or you may use raise hand feature in Zoom chat. Thank you very much. And for the next sections, I will give the time to Dr. Sylvie Ikawati. So for Dr. Sylvie, the time is yours.
Thank you, Habida. You can hear my voice here. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon all. I am Sylvie Kawati, the moderator for session 2 of this seminar today. So for the session 2, the topic is about plant pests. We have two expertise here. They are Dr. Ahmad Rizali and Professor Abdurrahman Saad Al Dawood. So before the presentation starts, let me tell you how the presentation will go. First, the speakers will be invited to present the material. First is Dr. Rizali, and the second is Professor Al Dawood. So for our third speaker today is Dr. Ahmad Rizali. Let me greet him first. Assalamualaikum.
Assalamualaikum. Bisa dengar suara saya?
Yes, we can hear your voice clear, Dr. Rizali. Okay. Okay. Before start the presentation, I will read the Dr. Rizali curriculum vitae first. So he is a full-time lecturer in the Department of Plant, Pests, and Disease, Faculty of Agriculture, University of Brawijaya. He is also the head of the Agricultural Entomology Study Program. And Dr. Rizali is an entomologist with the expertise in the study of ant, or we call myrmecology. He is also interested in the study of ecology or agroecology, especially biodiversity, landscape ecology, and biogeography. Currently, Dr. Rizali is also working on the survey, identification, and management of exotic invasive species. Now, allow me to welcome the speaker 3 today, Dr. Rizali, to make his presentation for 30 minutes. So, Dr. Rizali, please, time is yours.
Terima kasih, Bu Sylvie. Suara saya bisa didengar ya?
Jelas sekali, Pak.
Oh, jelas. Baik, terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. I'm sorry, maybe you will hear the background sound because in my area now it's very hard raining, and I hope the electricity is still on during my presentation. Okay, first of all, I would like to thank you. I'm honored to be here, to be a speaker among the professor from King Saud University. I'm not professor yet. I hope, yeah, I could be a professor maybe next several years. Maybe. Yeah, actually my topic or my presentation, yeah, maybe it's quite different with the previous speakers, yeah. And my presentation, actually I have presented in Bengkulu, yeah, last year, end of year. It's about the management of agro-ecosystem, yeah. And I think due to this topic is still interesting, yeah. So I think this topic is still important to be shared and discussed to all of you in this afternoon international seminar. Okay, let me share my presentation on my slide. Okay. Can you see this presentation or slide presentation?
Yes, this we can see your slide, Dr. Rizali.
No, can you see this slide or presentation? I'm sorry.
Yes.
Yes,
Pak
we can.
Rizali.
Yes,
Oh,
we
yeah.
can. We
Okay.
can see your
Yeah,
present.
thank you. I'm afraid that the presentation is still not
But please
coming.
make it slideshow, Pak.
Oh ya, saya sudah munculkan slideshow ini. Ternyata masih belum muncul slideshow-nya.
Belum muncul slideshow-nya, Pak.
Yep. Present already, Pak Rizali. Good.
Oh, sudah ini, sudah slideshow-nya sudah.
Go back again.
Oh,
Not yet.
like this.
Kan, Pak. Okay, yep.
Okay. Okay, thank you for your confirmation. Yeah, this is my topic. Yeah, that I would like to share to all of you about the research. Yeah, on the insect diversity in agro-ecosystem at landscape level or landscape scale. Yeah, that maybe is still rare research in Indonesia. Yeah? So I would like to share and discuss about this topic, especially about the utilization and prospect about this research, yeah, and also about the prospect or opportunity, yeah, how to do the research like this. Yeah, actually in developed country, yeah, such as Europe or US already done a lot of research like this, yeah, about the how to relate, how to consider about the landscape scale, yeah, to manage the biodiversity and agro-ecosystem, yeah, not only focus on the field or local scale, yeah. Because if we only focus on local or field scale, yeah, sometimes it's not will get the good result, yeah, especially related to the management of pests in agro-ecosystem. Okay, this is the outline of my presentation. Actually, first I will give the introduction about this topic here, and also share about the several research related to the insect diversity or insect communities, yeah, in agro-ecosystem, and also about the management of agro-ecosystem at landscape scales, yeah, and also about the utilization and prospect related to the research at a landscape scale. Yeah. Okay, this is the introduction. Yeah, I think everybody already knows, yeah, about the importance of biodiversity, yeah, in agro-ecosystem, yeah, including insects in this case. Yeah. That insects have the important role, yeah, in our agro-ecosystem. Yeah. So insects not only as a pest, yeah, but also more than 90%, yeah, 90% have the important role and as a important ecosystem services, yeah, in our ecosystem. And I think everybody also know about the hypothesis, yeah, why we need to conserve the biodiversity and agroecosystem, because related to this hypothesis, yeah, that stated by Elton, yeah, 1955 about the diversity stability hypothesis. Yeah, he said that if more, I mean, the higher diversity in agroecosystem, so we'll have the higher stability, yeah, stability of ecosystem services. It means that if we want to develop the sustainable agriculture, for example, in our agroecosystem, yeah, we need to increase the diversity in our ecosystem based on this hypothesis. Yeah. So this is the hypothesis that has a background for the importance of maintaining biodiversity in our agroecosystem. Yeah. Recently, we have the main problem, yeah, about the conserve or maintaining biodiversity in our agroecosystem, yeah, is affected by agricultural intensification, for example, pesticide application, yeah, that as a key or main factor that affecting the biodiversity loss in our agroecosystem or our ecosystem. Yeah. This is the problem. And that's why if we discuss about the conserving biodiversity in agroecosystem, sometimes we will have like a trade-off, yeah, between in one side we want to increase our yield, yeah, or crop yield, but in another side we want to achieve the sustainable agriculture. Yeah. So this is maybe you can write this paper. I think it's very important paper related to the concept that proposed by Palan. Yeah, Palan is a small paper, maybe more than 10 years old, about the minimizing the harm to biodiversity. Yeah. He proposed about the two concepts, yeah, but to minimizing the effect of to the biodiversity related to the management of agroecosystem, yeah, by use the land sparing, yeah, or wildlife friendly farming. Yeah. The difference between land sparing and wildlife friendly farming actually about the how to manage the area of the agroecosystem. Yeah, land sparing focus that conservation area should be protected, yeah, while agroecosystem can be utilized by using the intensive agriculture or intensive system. Yeah. I'm sorry. But for wildlife friendly farming, mean that we use the management, yeah, by wildlife friendly, that friendly to the wildlife or friendly to the biodiversity. Yeah. But as a consequence, yeah, if you use by wildlife friendly farming, you will get not high yield, yeah, compared to the intensive system. Yeah. So this is like a trade-off. Yeah. Nah, as I already mentioned about the trade-off, yeah, between high biodiversity and high yield, yeah, or high crop yield. Yeah. There are actually a lot of paper discuss about this. Yeah. Because this is actually a lot of researchers discuss about how to conserve biodiversity when the yield is low. Yeah, this is I think the trade-off. Yeah, so I think a lot of researchers, for example, Clough in 2011 and also Wurst, yeah, in 2022 already discussed about this. Yeah, and give like a solution. Yeah, about how we can combine, yeah, between high biodiversity with the high yield, yeah, for example, yeah, because this is like a trade-off, yeah, because usually if we conserve the biodiversity in our agricultural system, we will get a low yield, yeah, but based on these two papers, yeah, reveal that we can still get the high yield, yeah, if we conserve the biodiversity in our agricultural system. Yeah. Nah, why I give the introduction like that? Yeah, because if we want to manage or maintain the biodiversity in agro-ecosystem, yeah, based on the hypothesis about the diversity stability hypothesis, yeah. So it means that the management of biodiversity in agro-ecosystem, yeah, to control or to manage pests, yeah, in our agro-ecosystem. I'm sorry, I'm ecologist, so this is the ecological approach, yeah, ecological approach to manage the pests in our agro-ecosystem. So usually we only focus on or at field scale, yeah, or sometimes other people also give like a local scale, yeah. So field scale or local scale, yeah. But for the landscape scale, yeah, I like to be ignored, yeah. So this is the problem, yeah. Because if we ignore the landscape scale, yeah, and only focus on field scale, yeah, it will be problem, yeah, to manage or to conserve the biodiversity in our agro-ecosystem, yeah. Actually, a lot of people have, research, yeah, that reveal this or our statements, yeah, but to manage or to maintain biodiversity, yeah, not only need focus on the farm or field scale, yeah, but also to the landscape scale, yeah. And based on this analysis, yeah, or opinion by Changke in 2021, yeah, he proposed, yeah, he discussed that the diversification, yeah, diversification of this, for example, farm field, yeah, or in landscape term called patch, yeah. Patch is maybe we can call it like a field, yeah, field size, yeah. Can increase, yeah, can increase the biodiversity and also the crop yield, yeah. So that is I think very interesting, yeah. And also the mosaic, yeah. Mosaic means that you know that the mosaic of the the type of patch, yeah, of field or crop planted in the agro-ecosystem, yeah. Also important factor, yeah, or key factor that can increase biodiversity in agro-ecosystem. So this means that this is like evidence, yeah, that if we want to manage, yeah, or to maintain the biodiversity in our agro-ecosystem, yeah, so not only focus on the field scale, yeah, but also at the landscape scale, yeah, is on this opinion, or also the other research. Yeah. I will show the the evidence, yeah, and on the next slide. Yeah. Okay, the next I will discuss about the, maybe this is like a literature review, yeah, based on our research, yeah, because I also have done several research related to the insect diversity at a landscape level, yeah, with my student also. And also I will share about another publication, yeah, related to the insect diversity at a landscape level or landscape scale. Yeah. Okay, actually a lot of research, yeah, about the effect of landscape scale on insect diversity, yeah. And as I also mentioned before that research on insect diversity in agro-ecosystem, yeah, are not only affected by the field or local scale, yeah. Field or local scale, like for example, if we I'm sorry, if we planted refugia, for example, yeah, or flowering plants surrounding our crop plant, for example, is we call it field scale or local scale. Yeah. But if we consider about the minutes on the wide area, yeah, and consider maybe the different patch, yeah, different field within the landscape, yeah, we call it landscape scale. So with a lot of research, yeah, but I think in Indonesia is very rare, maybe, that already relate between the landscape skill, effect of landscape skill to the insect diversity. For example, Poor in 2003 or Changka 2007 is very old research, I think, that get the evidence about the effect of a landscape structure on insect diversity, on the biodiversity in agro-ecosystem. It means that if we want to conserve the biodiversity in agro-ecosystem, so we need to consider about the landscape skill. I will discuss later about the trait about the management of agro-ecosystem at a landscape scale is not easy compared to the management of biodiversity at a field scale. Yeah, this is the concept of the landscape structure that maybe some of you not understand about the landscape structure. So both of composition and also configuration of landscape in agro-ecosystem will affect to the to the biodiversity. So if we want to manage our agro-ecosystem with the landscape scale approach, so we should consider this composition and also configuration. This is the maybe diagram that show about the difference of composition and configuration. You can see here that for example for A, this is if we have two different patch, two different field. Maybe for example this is the paddy, for example, and this is for maize, for example, or corn. So it means that this is the heterogeneity is low compared to this the C. You can see this is the C. This is more heterogeneous than A. In addition, for the configuration, if we compare B and D, so this is the configuration heterogeneity. So this is we consider about the size of the field, about the shape of the field, and diversity of the plants. You can see that the B is more heterogeneity is more I mean higher than A, and also D is higher than C. So this is the difference between the composition and configuration. So if we do the research about the landscape, the effect of landscape to the biodiversity, usually we consider these two different type, but the composition of crop or field, for example, also the configuration. I think you can in detail, you can check in this paper, that written by Fahri in 2011. This is the example, that in the real world, for example, that if we want to study about the effect of landscape to the biodiversity, we will need to do like the real world that we can get from the Google Map, for example, from the satellite image. Also for another image method. I'm sorry. And we can convert to the vector, for example, like this, and we can test or we can analyze about the effect of the like this heterogeneous landscape to the biodiversity. And based on the previous research that I already mentioned, so the heterogeneity of the landscape will affect on the biodiversity on our agro-ecosystem. So this is the basic why we need to consider the landscape, especially landscape heterogeneity. You can see here that the x-axis actually is about the different configuration between landscape in one landscape, for example. So the green one means that only one type of plant, for example, or crop plant, for example, this is only paddy. While in in the second landscapes only covered by maybe maize, for example. Yeah. But in the other landscape, a lot, maybe four different crop plant in here. So based on the hypothesis, yeah. So only biodiversity that associated with this plant will exist in this type of landscape, yeah. Also in the second landscape. But if we compare to the complex landscape, yeah. So in this complex landscape, we'll have a lot of biodiversity. For example, this is species that found in cropland 1, cropland 2, 3, and 4, etc. So this is the concept, yeah, or the hypothesis about the importance of landscape heterogeneity, also the landscape composition on the biodiversity. Yeah. So based on this concept, actually, if you want to manage the landscape of agroecosystem, sorry, of our agroecosystem, if we increase the diversity or heterogeneity of patch, yeah, in our landscape, so they will increase the biodiversity in our agroecosystem. Yeah. Actually, a lot of research, yeah, reveal about the effect of landscape. For example, this is from Steckel that found the effect of landscape type to the, for example, this is the bees, yeah, and also wasps, yeah, significantly affect this diversity. And also you can see here that effect of the presence of, for example, semi-natural habitat. Yeah, you can see here. Maybe this is the picture is small, yeah, but the dark green, yeah, this is the like a semi-natural habitat, for example. Yeah, so this composition significantly affect the biodiversity, yeah, and also shape the ecosystem services in the agroecosystem. Yeah. And this is my student, yeah, my doctoral student also study about the effect of agricultural composition related to the biological control, yeah, in agroecosystem. Yeah. So she got that the landscape composition, yeah, significantly affect especially the number of patch. Yeah, I will discuss later. This is very interesting research because number of pets, even though the monoculture system, yeah, but if they plant it in the small patch, yeah, a lot of small patch of the same species or same crop plant, yeah, will affect to the, or will support the presence of the natural enemies, yeah, in agroecosystem. What about landscape at the agroforestry system? Yeah, as you may know that the agroforestry system can support the biodiversity in agroecosystem. Yeah. The question is, what about the landscape? Yeah, within the agroforestry system. Yeah. Based on this research, yeah, by Boss in 2007, yeah, he found that he did in the Kakao agroforestry system, yeah, in Sulawesi. He found that the Kakao agroforestry system has important contribution to the conservation of ants or insect diversity, yeah, in Kakao agroforestry system. Yeah. But what about the effect of landscape, yeah, in the agroforestry system? Yeah. My student and I study about this, and we found that even although the agroforestry system conserve or maintain the biodiversity, yeah, in agroecosystem, but still, I'm sorry, still, this system is also affected by the landscape in this agroecosystem. Yeah, this is my student, yeah, that also study this about the effect of landscape at agroforestry system, yeah, that also affect the biodiversity, especially to the natural enemies, yeah, in agroforestry system. Yeah. It means that even though we call it for local or field skill, yeah, that the agroforestry system has important contribution to the conservation of biodiversity in agro-ecosystem, but at a landscape level, yeah, I mean the agroforestry system still affected, yeah, by the landscape surrounding the agroforestry system. Yeah. Okay, what about the management of agro-ecosystem at the landscape scale? Yeah. So we have discussed about the research, yeah, related to the insect diversity at landscape scale. Yeah. Actually, there are several opinion or discussion that already discussed by the researcher. For example, I put this publication by Chanke, but because he discussed about the organic farming, yeah, but also discuss about how we can design, yeah, about the biodiversity friendly landscape. Yeah. So not only at a local or field scale, yeah, but also biodiversity friendly landscape. Yeah. So he discussed about this. I think you can read directly to this paper, yeah, about the different management, yeah, at the local scale and landscape scale to conserve or to maintain the biodiversity. Yeah. For example, like this is the concept actually, but still need to be tested, yeah, to conserve or to maintain biodiversity in our agro-ecosystem. Yeah. So the local scale, usually the farmers will focus on the diversity of crops, yeah, or maintain traditional agricultural practices, for example, yeah, or minimize pesticide. Yes. This is the local scale. Yeah. But for the landscape scale, yeah, you can see that the landscape scale, we need to increase landscape complexity that I already discussed about the configuration and composition, yeah, of landscapes is part of the landscape complexity. Yeah. Also, if the landscape already simple or simplifies in the wide monocultural system, yeah, he suggests to restore the simple landscape to the complex landscape, for example, yeah, and so on. Yeah, you can direct this because I think the time almost up. Yeah. Okay. So how we can diversify the landscape scale? Yeah. Actually, there are several concepts. Yeah. And maybe we need to discuss later about because management of the agriculture at landscape scale is not easy. We need to include the policy makers, for example, yeah, or we include another landowner, for example, because as you know that the farmers in Indonesia, yeah, is on the data from the BPS, yeah, data from the BPS that the average of the landowner in Indonesia is about 1/4 hectare. Yeah. It's very small. Yeah. So if we want to manage, yeah, or to design, for example, at a landscape scale, for example, we need to include all of the farmer within the landscape. Yeah. And one of the approach, for example, for diversification, yeah, by providing semi-natural habitat, for example, yeah, to increase the source, yeah, for the biodiversity, the natural habitat, and also increasing the diversity of crop plants. Yeah. So this is the approach that we want to increase biodiversity in our agro-ecosystem. Yeah. Last, I want to discuss about the utilization and also prospect, yeah, about this kind of topic or this kind of research. Yeah. So actually the management of insect diversity, yeah, in agro-ecosystem need not only field or local scale, yeah, but also need the landscape scale. Yeah. This is we must consider about this. Yeah. And also the management of that I already discussed before. Yeah. because this is the landscape scale, we need to to get the support from the regulation maker or policy makers to implement this concept. And also this is I think like a prospect for our our research topic, for example, to test because some of research actually only done have been done in the several geographic area. So we need to do like another research in different geographical area to get more evidence about the management of biodiversity at a landscape level. This is the research that have been done by German researcher in Jambi. I think this is also good idea that maybe we can develop like this. This is actually in oil palm plantation. You can see here that how because the problem of monoculture actually problem of monoculture in the wide or big area will decrease the biodiversity and like supported the presence of pests in oil palm plantation. This is the problem. So the concept of this research actually the biodiversity enrichment experiment it's called effort. They in how if in the oil palm plantation increase the diversity of another plants. So you can see here that the diversity level different in one plot, so different crop plant and also different size of patch. So how is effect to the biodiversity? Actually not only to the biodiversity but also to the income to the farmers. For example, if we planted in here the fruit crops or fruit plants, so we'll get another income, besides besides from the oil palm itself. So I think the idea is interesting, but still need to do the research to get the more evidence. Also, this is the another research by UK researcher that they also actually are almost similar, but this is the different consider about the effect of age effect and also the core habitat to the biodiversity and also to the yield of the oil palm. Okay, maybe I skip this. This is actually the recent concept that proposed by Marini. I think also interesting to study. But what about if we compare a small field to the with the wide field or big field to the biodiversity in agro-ecosystem related to the different landscape composition. You can see here that this is the concept actually, because every farmer, especially in Indonesia, this is for professor in King Saud University, because we only have small field usually in average. So what is the effect of size, field size to the biological control in agro-ecosystem in related to the landscape scale? So this is, I think, interesting to study. And this is actually my study, my previous study in at oil palm plantation. I have submitted to the Insect Conservation and Diversity. I found the interesting result that even though the oil palm is a monocultural system, monocultural system in very wide area. For example, I did this research in Central Borneo. So I found that the composition, you can see here that this is the landscape composition. So even though the oil palm is monocultural system, but due to the farmers, planted in the different patches, which different patches have different age, tree or tree age. Yeah. So this also affect to the biodiversity in oil palm plantation, yeah, and also affect to the natural enemy communities, yeah, in oil palm plantation. Yeah. Maybe if we already accepted, I will share to to you. You can check in the website. Yeah. So it means that the management of agro-ecosystem at landscape level is very important. Yeah, even though in a monocultural area, in the wide monoculture area, yeah, because in oil palm plantation, yeah, they have different age, yeah, different landscape composition. For example, in some some landscape have remaining forest, for example, yeah, but in some landscape fully only oil palm plantation, yeah, they have the different natural enemy communities. Yeah, it means that the different agro-ecosystem or different area, yeah, will have the different condition, yeah, or landscape condition that may support to the natural enemy. But still, the presence of the semi-natural habitat, for example, is still important. Yeah, that's why regulation related to the oil palm plantation farmers, yeah, they should protect or conserve the HCV, for example, high conservation value, yeah, area in oil palm plantation that can support or manage the biodiversity in oil palm plantation area. Yeah. And I think this concept or these findings, yeah, is also applicable to the another plantation, even another seasonal crop. Yeah, for example, paddy that also in Java planted in the wide scale area. Yeah, but if they planted in different patch size, for example, yeah, in different complex agricultural complexity, they will support the natural enemy communities in agro-ecosystem. Yeah. Okay, that's all. I'm sorry if the time is up. Yeah. I hope you get the idea about the effect of management at a landscape scale, yeah, to the biodiversity, especially to the biological control agent, yeah, in agro-ecosystem. Yeah. I think that's all. Thank you very much for your attention. I give back to the Bu Sylvie. Monggo, Bu Sylvie. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Rizali, for giving such an informative and interesting presentation. So I think the presentation is precise for the time. So for the fourth speaker, because our time is limited, is Prof. Abdurrahman Saad Al-Dawud. So before he start the presentation, I will read for the curriculum vitae of him. So he is a full-time lecturer at the Plant Protection Department, College of Food Science and Agriculture, King Saud University, Saudi Arabia. And his area of study is economic entomology, with the main research is on integrated management of dead palm and forest pests. Besides studying bioecology and management of insect pests, Prof. Al-Dawud has also conducted insect surveys in various geographical regions and their identification, as well as species level taxonomic revisions. For instance, he has studied entomofauna of Western Forest region of Saudi Arabia and large-scale discovery of new taxa and the analysis of diversity and distribution pattern of ant in Africa and Madagascar. So before the presentation start, I will greet him first. Prof. Al-Dawud. Halo,
Yeah.
Assalamualaikum.
Waalaikumsalam.
Okay, so now allow me to welcome the speaker for Prof. Al Dawood to make his presentation. So Prof. Al Dawood, please, time is yours.
Okay, thank you very much. Assalamu alaikum wa barakatuh. I'll try my best to summarize what we're doing here and what is Saudi Arabia and what is the King Saud University and what is the College of Food and Agriculture Sciences and our department, Plant Protection, and some activities of our unit, which we call Economic Entomology Research Unit. I titled my talk Greening the Desert. And in reality, we've been talking about this before, more than 10 years ago. Now it's in process and it is active. You can see the 4 pictures that represent some parts of Saudi Arabia. Of course, everybody thinks of these 2 holy places when he thinks of Saudi Arabia, Mecca and Medina. And I first say to all of you, because we are now in the afternoon, I say good afternoon. Salamat sorry. I was planning to say Salamat, what is morning? Pagi, but we passed nearly 1 hour. Now, Saudi Arabia has many diverse areas. We are known for, let's say, sand and camels, and the mud houses on the bottom left. Now trains are working.
Hello,
Saudi Arabia has been transferred to what you can see right now and more. It's going to be more than that in the coming 10 years when the Vision 2030 is completed. A lot of islands will be created and inhabited either through tourism or inhabitants. A lot of big cities are going to be international all over the world. Riyadh is now expanding. 8 million people will become 15 or 20 million people in the end of 2030. I know it's nothing compared to Jakarta and Indonesia, but it's big for an Arabian country like Saudi Arabia. A lot of adverse conditions can pass to the Saudi condition. Snow, yes, we have snow in the north, and we have a lot of greenery areas in the west, and many activities on other parts of the kingdom. A lot of universities, 3 that are famous in science: King Saud University, Princess Noura bint Abdurrahman University, which is ladies only university, and King Abdullah University for Science and Technology. The greening desert is basically targeting changing this desert sand into green land. The objective is to plant a lot of trees and shrubs and grazing plants, and in the same time to preserve the living organisms and plants in the natural habitats. Before 160,000 years, Saudi Arabia was green. Muhammad sallallahu alaihi wasallam said, Yawm al-Qiyamah, the day of judgment, never come back until the Arabian Peninsula become green. Topography of Saudi Arabia is so diverse, as I said before. This is a map of Saudi Arabia, and we have sand, of course, and sand and water, seas, mountains and valleys, high mountains in the west, and a lot of greenery in the west and northern part of Saudi Arabia. A lot of topography and changes and weather conditions, different forms of land in the kingdom, and a lot of studies have been done to test the soil and know the soil kinds of Saudi Arabia. So what are the issues in the soil desert? We have, of course, water, and then thinking of greening that desert, and then we have to support a lot of modern technologies to overcome any problem that is expected when we plant this big, huge desert. So what we're doing actually is we're using technology, as you see on the top right, and then learning about the sand then finally creating green land through agriculture, or making the nature come back again by preserving it. A lot of beautiful dates or date palms are present in Saudi Arabia, but in the north, there are a lot of fruits like olives, and we are competing internationally with our olive oil and olive or table olives. So date palms in Saudi Arabia, Muhammad talked about dates because Allah subhanahu wa ta'ala talked about dates. And talked about dates when he was talking about our mother Mary, when she was delivering Jesus, under the palm. And that's a famous story, and it's read in the Quran. So we have more than now 32 million date palms in Saudi Arabia. A lot of issues are related to this palm, from the propagation until the protection of its part, all of its part, specifically, and I'll tell you why, the trunk of this tree. 34 years of date production in Saudi Arabia. Now we passed 1.3 million tons per year as production, and huge areas are expanding of dates. A lot of beautiful date varieties like Ajwa that you all know on the top left, but we have more than 60 to 100 famous dates in Saudi Arabia, and it is awaiting a lot of exports to the whole world. A lot of processes that are going into making these beautiful dates into many different shapes and products. Now, what are the problem of pests in Saudi Arabia? For date palms, if we think of date palms, and I want to concentrate on that, we think of the red palm weevil. Of course, in Indonesia, you have 4 species of the Rhynchophorus, but our species is Rhynchophorus ferruginus. You have 4 of them in Indonesia. You have threat for the oil palm and for other palms in Indonesia. And I'm glad that I'm working with my previous students who are now doctors in the universities in Indonesia, and I have this doctor, Dr. Koko Sutanto, along with my research support teams in our team. So red palm weevil is a big problem that came from the west or the east of Saudi Arabia, possibly from India or other places in Asia in 1985 that started. And from there, it went all the way to the Americas. So it is a very serious pest. I don't want to talk about it all, but I want to tell you one thing, that Indonesia and Saudi Arabia are included in the distribution of this insect. These are some of the symptoms of this insect. Finally, that it will knock the tree down if it is not followed. Now, let's talk about the research worldwide. This is a 2016 record where you can see Saudi Arabia is among the top producers of research regarding red palm weevil, and Indonesia at that time is 3 papers. At least 2 papers we've done after that with the doctors that have been graduates of King Saudi Arabia, and they are now working in Indonesia. About 17 papers we have published so far in red palm weevil. 6 extra ones are in the wait for publication just in the topic of red palm weevil. Now, there is a possibility to apply IPM, which is integrated pest management, to manage the red palm weevil and other insects also, of all kinds of crops. But let's concentrate on red palm weevil, that there are successful examples using all the tactics allowed or can be used in managing this red palm weevil. That it takes sometimes, of course, but we can finally get to zero infestation of such areas. This is one of our challenge, is to communicate with the world. We were working and like this international seminar in Gajah Mada, and we looked at the dates in Jawatenga, and even we looked at the dates possibilities planted of Barhi, for example, in what's the big island? Kalimantan, okay? So let's talk about our team. I want to introduce you to my team. Many of them are doctors already. Most importantly, top left, Coco Santanto. This is his picture. He graduated his PhD in 22. Dr. Alan Sofan here on the right, and Dr. Sikerno on the left. I am so proud that I had these students in my lab, among other students from all over the world. So what is my target? Is foreign collaboration. You can see that I am collaborating with Dr. Alan Sofan and Dr. Sikerno. Dr. Coco is with me now. And this is in University of Gajah Mada and other countries and universities. Now, I am looking really seriously of working all the plant protection department here with anybody interested in Brawijaya University in Indonesia. Thank you very much, but I'm going to give you another presentation if you allow me. How do I stop this and go another one? Hold on. How do I stop the share? Here, where is it? New share. New share. Okay. Just to give you a background about our activities and what we are doing. How many minutes I have left until 1:15? Huh? Yeah, until 1:15. How many? Until 1:15. How many minutes left? 14. Okay, I have 14 minutes. Thank you very much. I'm going to talk to you about our ongoing research and past research in ERO, Economic Entomology Research Unit. This is my team again. Our vision, like anybody, we need global recognition for quality research. Our mission is to do scientific research of an international standards and establish a scientific center to meet the local challenges. We have published more than 200 papers with ERO, all our activities so far. Now, what is our agenda? We're talking about using of modern biotechnology approaches and exploring secrets at molecular levels and using the DNA barcoding and biogeographic studies for studying animals and plants, establishing quality and excellence in teaching and learning, and capacity building of our members and other members interested. And we need to continue international collaboration and contribute to the scientific community and provide innovative solutions. So we are interested in red palm weevil, date moth, cotton leafworm, ants, termites, and rice BPH. So RNA biotechnology we have used in different aspects, but we have done the silencing of the protein genes in red palm weevil, and we were successful to make eggs unhatchable. And the same thing we did with the odorant receptors in the same insect, red palm weevil, where we made the insect not be able to smell the ceremon of the plant, which is date palms. And some just information of how we did it. I don't want to go details in that, but again, we use now microbial biological control to manage insects. We use baculovirus and fungus against the insect, cotton leafworm insect, and we use again the same fungi and nematodes against red palm weevil, and we use it an injection in the field, and it is as successful as any pesticide, but safer. We have used barcoding to differentiate between red palm weevil. We use samples from Saudi Arabia, from Indonesia, from Pakistan. This is the work of Dr. Sikerno, and we have found very interesting information regarding to that. In the control of Ephistia cotella, which is the date worm, we have done the same thing. We silenced the gene that is responsible for the egg protein and made the insect not hatch its eggs. The microbial control, as I said before, we used viruses against cotton moth, and it was successful to manage the insect. And the barcoding, as I mentioned before, for the red palm weevil, and now for the ants. And we have established a lot of collections from all over the kingdom and other countries and compared using barcoding, and it was very nice work that is published. Termites, same thing that we have done with ants. We are using with termites, collected a lot of samples from Riyadh region and done a lot of barcoding to test the presence of these species morphologically and molecular aspects of the identification. And we have published work on these issues also. A lot of work, a lot of papers, and a lot of books, but the most important thing is the insect that is in Indonesia, which is Rhynchophorus vulneratus. You see different colors. Dr. Sikerno worked on that and had a good publication published after his dissertation and before that. Again, the second species in Indonesia, and then in Pakistan. That's all for now. I don't want to take longer time, and I'd rather keep it for answer question session. Thank you very much.
Welcome, Prof. Al-Dawood. Thank you for giving such an informative and interesting presentation. So for the next session for this seminar is for question and answer session. So in chat room, we have some question for Dr. Rizali and Prof. Al-Dawood. So for first question is for Dr. Rizali. From Nurfianto Giar Pangindung. He have two questions here. So first question is, in field scale biodiversity managements like planting flower plants, how far the range of influence the flowering plant on landscape scale in the agroecosystem biodiversity? And for second question is, how about landscape scale biodiversity management in urban agriculture for IPM? Okay, Dr. Rizali, are you ready to answer? You get the questions?
Ya.
Okay.
Bisa dengar saya, Bu Sylvia? Bisa ya? Okay. I would like to answer the question from Pak Nurfianto. Yeah. First of all, I would like to thanks for the. I think it's very innovation. Related to the management of field skill and landscape skill. As I already mentioned that why we need the field skill, but also a landscape skill, because skill sometimes cannot cover.
Not good voice, Prof. Not good voice. Your voice not so good, you know.
Yeah.
Seems good.
Okay. So because we need not only field skill, but also landscape skill, because sometimes, or not only sometimes actually, but field skill is have a limitation, like a distance and area to cover, that you ask in this question, that how far, for example, the field skill, if we planting the flower plant, to that can cover the biodiversity in our field, in our crop plant, for example. I think this is a common question, that how far, but actually it's depend on the taxa, or depend on the natural enemies or biodiversity that we want to conserve or to study. If we, maybe you have heard about the threat, effect of threats, effect of threat to the the species composition and in agricultural field. Yeah, if the big, for example, the big species or the compared to the small species, small insects, for example, with the big insect, they have the different different range, yeah, different strains that can access to the flowering plant, for example. Yeah, that's why it's really depend on the group or taxa. They can access to the flowering plants. That's why to conserve the biodiversity in agroecosystem, yeah, we need not only the field skill, yeah, but also the landscape skill. We hope that we can cover, yeah, the biodiversity not only based on the distance from the refugia or flowering plant, yeah, but also from the landscape composition or configuration that also will affect the species composition in agroecosystem. Yeah, that's for the first question. And then for the second question about the landscape management, yeah, at urban agriculture. Yeah, as I also discussed in my presentation, yeah, this is the very big challenge, yeah, that as you mentioned that in urban agriculture, yeah, the problem is that the how we can do the collaboration, yeah, between the landowner. Yeah. This is the big challenge or the big problem. Yeah, that's why I or not only me, a lot of ecologists, yeah, propose that we need to give like a scientific background and evidence to the policymaker, yeah, that because they are the most important important person that can give like a decision, yeah, about to how to manage the biodiversity at a landscape scale in urban area, yeah, for example. Yeah. So it's not easy, but needs like a political approach, yeah. And this is the challenge actually to manage the biodiversity in urban agriculture. That's all. I think. I hope I can answer the question. I give back to the Sylvie.
Thank you, Dr. Rizali, for the answer. Maybe Pak Nurfianto, there is a feedback or doubt clear? Okay, with Pak Nurfianto, maybe we go to next question for Dr. Rizali for from second participant from Faiz, yeah, a student from University of Brawijaya. Dear Dr. Rizali, what do you think about the correlation between increasing settlement patch in a landscape and climate change? And is the relationship between these two threat to biodiversity? Thank you.
Yeah, thank you, Mas Faiz ini ya, for the question. I think it's very also interesting question, yeah, related to the relationship between composition of landscape, yeah, to the climate change. Yeah, actually, it's still in debate actually about the climate change. I think you can try to read the effect of climate change to the biodiversity, and also in relation to the habitat fragmentation. Yeah. So habitat fragmentation also very interesting to be discussed. Yeah. That for example, the transformation of land use, yeah, or land use transformation based on the research will affect to the climate change. Yeah. And also this means both we call it global jeopardy, yeah, So double jeopardy means that from the land use change back to the biodiversity. In addition, from the climate change also affect to the biodiversity. So this is like a double jeopardy of the both of land use change to the and climate change to the biodiversity. You're right that this is a very big threat, yeah, to the biodiversity, and this is a very big challenge to the ecologists, yeah, how we can conserve the biodiversity, because the two important factor, yeah, affected the biodiversity in agro-ecosystem is the first about the land use proposed by Foley. I think you already know about the Foley, and 5 discuss about the effect of land use change to the biodiversity. And also the second is about the agricultural intensification, yeah, and also the third about the climate change that will affect to the biodiversity. I think that's all. I answer the question from Mas Faiz ini.
Okay, thank you, Dr. Rizali. Mas Faiz is already clear? Okay, because he is offline now, so he said not clear. So for the last ending for Dr. Rizali, what factor from Pak Syarif Hidayat from UNPAD, what factors can determine that biodiversity is beneficial for plants? Thank you.
Yeah, actually there are several hypotheses, yeah, related to the relationship between biodiversity and stability. I mean the stability of ecosystem, including the beneficial for plants. Yeah, you asked about this. Yeah. So, but the main I call it the main, yeah, the main hypothesis that people agree is about the diversity stability hypothesis. Yeah. It means that because this is analog from the natural forest, yeah, or natural habitat, yeah. This is analog from this hypothesis actually come up from the from the natural forest or natural habitat condition, yeah. That we know that in natural habitat or natural forest, yeah, because in natural forests have the high diversity of vegetation or plants, yeah, so they have a high diversity of biota that associated to the plants, yeah. So with the higher diversity associated with the plants, with the higher plants, so it will create like the stability. That's why this called diversity stability hypothesis. Yeah. So if you ask about the factor that determine the biodiversity, actually the habitat condition, yeah, that I also have answered the previous question. Yeah. For example, land use change, yeah, or fragmentation habitat, yeah, it's a very important factor that can affect to the biodiversity loss in the ecosystem or in agro-ecosystem, yeah. So that's all. Maybe I hope I can answer the question. Thank you.
Syarif Hidayat, that is clear? The answer from Dr. Rizali? Okay.
Okay, thank you. It's very clear.
Okay, thank you, Pak Syarif Hidayat. So for next question is 3 questions, I think, for 3, yeah. I think 3 questions for Prof. Al-Dawood. So for the first question, I think it's for Al-Dawood about the entomopathogenic agent. So from Amelia Sebayang, Ibu Amelia Sebayang, how to get persistent results to control pests using the entomopathogenic agents, because we know the infection of the entomopathogenic agent will be influenced by several factors such as environment.
Okay, you want to answer? Okay, this is the specialist on my left, Dr. Kuku, who is working on the application of the fungi, two species we have, Beauveria bassina and Metarhizium, right? And Metarhizium, along with the nematodes. Yes, if you are spraying it in our environment, it is very harsh. Of course, it's not going to work unless if you're doing it in specific seasons, like this season now, it is a rainy season. But what we're doing is the following, and this is going to answer two questions, this question and the following question, because I forgot to mention our efforts on the early detection of red palm weevil, which I think we are the first in the world doing that for red palm weevil, to early detect the insect, number one, to treat the insect, number two, with our methods, and number three, to make sure that the infestation is stopped. And we use this machine. I don't know if you can see it. I can show you here. You can see the background white here. It's a machine with a rod here that we drill the trunk, put it inside the trunk, and then we listen to the eating of the larva. This is also sent to the internet. We download it, we analyze it, we change the sound wave into digital numbers, where we can get the pulse numbers and the frequency numbers in decibels. And then we can know kind of exactly the level of infestation, whether it's low or medium or high. Now, how do we apply our treatments, whether biological or pesticides? We drill in a spiral manner, 4 drills 50 centimeters apart from down the bottom of the trunk up until the 1.5 meters. And we inject our treatment, including the biological agents, with special apparatus injection, we call it balloon, that is under normal pressure of the elastic, and it takes about 15 to 1 hour for whatever inside that bottom balloon to go inside the trunk. After that, we read before, we know exactly that this tree is infested, so we only treat those that are infested, and it is nearly as early detection as possible in the whole world. And after treatment, we can continue doing the readings dependent on our experiment, either daily, weekly, or monthly. Now we have more than 12 farms with more than 1,500 palms that we are following for the last 1.5 years after our treatment, and all these palms are safe and not infested anymore. So this is an answer that when we inject the fungi or the nematodes inside the trunk there is nearly 100% humidity. Temperature is stable between 24 up to 37 degrees, yes, all over the year. So it's not only that it kills the larva, we found that the fungi and the nematode can stay after that when there is no insect as an endophyte, meaning that tree is nearly immune against the new infestation of the red palm weevil. So I hope this will answer the first question about the persistent results to the control pest, and answer the first question of Ito Fernando, where he asked about the early detection of the infestation. Now, the last question or second question of Fernando, he says, is red palm weevil an endemic species? Yes. And I'm telling you, it is endemic now in Europe. You mentioned that EU it's a quarantine species. Yes, it is quarantine species all over the world, but they have it. Any date palm or any palm in the world has the Rhynchophorus genus with, of course, different species in it, from the Americas all the way to Japan. Versus not America, Japan, but America, go back all over the globe and come back to Japan. So it is all over the world. Whether you call it quarantine species or not, it is present. So yes, it is an endemic species, I can tell you, all over the world. Last question, do we eat the larva of red palm weevil? Few people here in Saudi Arabia and in some Arab countries, they eat locust. And we know in Indonesia, few people are eating larvae of red palm weevil. And we know in some areas in the Americas, they're eating the larvae or the grubs of some beetles. But we don't eat the larvae yet. We don't have this culture yet. Although I think it is like any larva of either lepidopterans, moths and butterflies, beetles, weevils. They are all edible and they are high concentrates of fats and proteins and other minerals and vitamins. But it's a matter of social acceptance of such issues. There's another question here, if you allow me, that says, do you have a Rheactis rhinoceros? Yes, we have it in Saudi Arabia. You know that this insect is not the pest of a healthy plant. It is eating as larva the manure of animals. But when there is bad irrigation and the water is touching a lot the trunk and there is decay in the trunk, then this scarabid beetle will infest that palm or that offshoot. Same thing that is going on in Indonesia. What else? And we are looking into this issue by reducing the cultural methods in the farm to reduce this insect, the scarabid, and the red palm weevil also, because red palm weevil is a wound parasite, meaning it cannot lay eggs on healthy, not cut, dry trunk or leaf of the tree. But if there is decay of water or there is wounds, there's going to be infestation. What do you say? Sorry. He paid attention to me. I'm not talking about the species of Rheactis rhinoceros. I'm talking about the species we have is Rheactis elegans. Sorry. Thank you very much, Dr. Cooper. Right. What else? Do I have any more questions? No more. Okay. The mic is with you, dear.
Okay, thanks, Prof. Aldaud, for the answer. Exactly, I have one question for Prof. Aldaud. I see in the PPT of your presentation for the technology in your lab project now is about nanoparticle. So I want to know what exactly the project.
We're meaning here applying the nanoparticles with the RNAi, the gene silencing. We are using, because we have done it on the organism in the lab by injection, and we have done it in the lab where we put our produced RNA in the feed, and the larva is feeding on it, and it's effective. And it's in the way of publication now. Our last task is to inject the RNA in a nanotechnology method inside the trunk. After we detect that it has the insect, we inject and follow the plant also by listening to the trunk and finding if the RNA used through nanotechnology will be effective in the tank of the plant. We're hoping it's going to work. And that's, we mentioned nanotechnology.
Thank you, Prof. Aldo, for the answer. I think so the technology in the Saudi Arabia is enough advanced for the detection and for the pest management, I think. So maybe there is one question more for the speaker. We still have around 7 minutes less. Maybe for Dr. Rizali, this is still one question for the participant. Dr. Rizali, please give your opinion as an ecologist for food estate program, especially for landscape management. Thank you.
Yeah, thank you very much, Prof. Norinda, for the difficult question. Yeah. Actually, this is the very difficult question because this program is very political decision, yeah, very high political decision, and I think Bu Norinda maybe more have answer about this program, yeah. But based on the study, previous study, that the food estate program is not achievable or feasible, yeah, to be done, especially a lot of ecological aspect that not be considered, yeah, by government, yeah. But of course, this is the political decision, yeah, that we need to give like a support or to give like input, yeah, to the who has responsibility to do this program, in especially in Kalimantan or in Sumatra, yeah, that they need to consider, yeah, when they design about the food estate program in there, and how to design the sustainable agriculture, not only focus on to get the high yield, yeah, based on the consideration, yeah. And this is actually the common problem in everywhere, that scientists and policymakers have the different consideration, yeah, about how to to develop the program related to the yield, yeah, in agro ecosystem, yeah. That's why I think the program from our government, for example, to make like an interface, yeah, interface between policymaker and scientists is very important, yeah, because the problem is that there are no interface, yeah, there are no interface between the policymaker and scientists, yeah. This is the problem, yeah. No interface. That's why we need to have like interface, so we can discuss and communicate, have a deep communication, yeah, with the policymaker, how to do like sustainable program, yeah, to get the food estate, for example, yeah. This is my opinion, but I think Bu Norinda or Prof. Norinda as a researcher, yeah, under the Ministry of Agriculture, more maybe more understand about this situation and condition, yeah. And even if we discuss about the who has responsibility to this program, is sometimes very strange, yeah. Why under the Ministry of Defense, for example? Sometimes it's very funny, but yeah, that's our country, that we have to support and also give the input. Yeah, as long as they can get or they can understand our our input or our argumentation. Yeah, so we hope they will apply or implement the concept to this project or this program. Yeah, I think that's my opinion, and I believe that Bu Prof Norinda more understand about this condition or this situation. I think that's my opinion. Thank you, Bu Norinda. Monggo. Bu Norinda lebih paham ini. Saya kembalikan ke Bu Sylvie.
Thank you, Dr. Rizali. Maybe Prof. Norinda will give feedback directly or opinion, other opinion.
No, it's thank you for your opinion, Pak Rizali.
Okay, I think we still have 1 minute, maybe because the time is up. So I think this the last of our session for this seminar 2 days. So I would like to thanks to Dr. Rizali and Prof. Al-Dawood for their informative and interesting presentation. And for Dr. Rizali and Prof. Al-Dawood, as a sign of gratitude from our university, we will give certificates for Dr. Rizali and Prof. Al-Dawood. This our sign of gratitude. Thank you very much. So
thank you,
maybe
Bu Sylvie and committee.
You're welcome, Dr. Rizali. Okay, maybe there are statements from Prof. Al-Dawood for this seminar or for other collaboration.
I didn't get you. Can you repeat, please?
Maybe there are statements for this seminar, Prof. Al-Dawood, for the last statement, maybe?
Yeah. Number one, I really thank you very much for this fruitful international seminar, that it is very usual through my experience of working with Indonesian universities. And I am glad that not only students and technicians, but also faculty members from universities of Indonesia and Saudi Arabia have passed through this seminar. And I heard that the Indonesian Agriculture Department are listening with us. And I'm telling you that the Agriculture Department in Saudi Arabia are with us here. And many of my graduate students and many of other department graduate students are tuning in to this seminar. So it is really a multi-international seminar that is needs to be given many thanks to your university for this great effort. Just one last thing, because some people who have joined from Saudi Arabia are wondering if there is a possibility that they can get certificate of attendance because they joined with us. They are filling, some of them, the questionnaire about the seminar, but it's just a question that needs to be answered. And I thank all that are behind such a successful international seminar. Thank you very much all.
Thank you for Prof. Al-Dawood. So finally, I return to Bu Hafidah.
All right, thank you very much. Thank you for the insightful knowledge from Dr. Ahmad Rizali and Prof. Abdul Rahman Saad Al Dawood. And thank you for Dr. Sylvie for leading the discussions. To end this event, I would like to invite the head of Plant Pest and Disease Department, Faculty of Agriculture, Mr. Lukman Kurata Aini, PhD, to deliver the closing remarks for this international seminar. For Mr. Lukman, the time is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good
Waalaikumsalam
afternoon.
warahmatullahi wabarakatuh.
To all Honorable Rector of Universitas Brawijaya, and Dean of the Faculty of Agriculture, Universitas Brawijaya, and President and also faculty members from King Saud University, Saudi Arabia, all professors as speakers in this seminar, and also Indonesian ambassador in Riyadh, Saudi Arabia, and all audience who attended this seminar. Alhamdulillah Rabbil Alamin. I would like to thank God Almighty because we can join this forum through the end of the seminar. And we have already know that this seminar is held as a forum for scientific communication for all stakeholders, including our students, lecturers, scientists, and general public, to update the latest knowledge about the development of plant protection science. And I also have joined this seminar through the end, and many interesting information and new insight on the advanced research about plant pests and disease that give us new ideas or topic towards how to manage plant pests and disease, particularly in two countries, in Indonesia and Saudi Arabia. And this will give any opportunities for us to develop more collaborations in the near future in terms of research, knowledge exchange, or maybe our students interested to take advanced study in Saudi Arabia and particularly in King Saud University and so on. Finally, on this occasion, I would like to express my deepest gratitude to all the speakers who shared their knowledge and experience with us. There's Professor Muhammad Ali Al-Saleh, Professor Yasser Ibrahim, Professor Abdurrahman Saad Al-Dawud, and Dr. Ahmad Rizali. And also we thank the Rector of Universitas Brawijaya, Dean of Faculty of Agriculture, Universitas Brawijaya, and also President or representative from King Saud University, and also Indonesian Embassy of Riyadh, Saudi Arabia, particularly to the Ambassador, Dr. Abdul Aziz Ahmad, and then the Deputy Chief of Mission, Mr. Arif Hidayat, and also Education and Culture Atas, Dr. Batur Soleh, who have kindly facilitated this international seminar. And finally, we also thank to the committee who have worked hard to hold these activities, and all audience who are willing to attend through the end of the seminar. Here in this meeting room also attended by students, particularly from postgraduate student of our department. And the attendance of this seminar today reached about 275 audience. And it's been a pleasure being with all of you today. We hope that this seminar can provide benefit and new insight for us to further develop plant protection, to crop security for sustainable agriculture, so that it will further increase the benefits for the entire community. And we can increase the collaboration in the near future. Thank you. Goodbye and see you in the next activity. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih. Thank you.
By the end of the closing remarks, the international seminar on plant protection is officially ended. Thank you so much for all of the wonderful keynote speakers. We are so grateful to have all of you here. And thank you so much for all of the guests, the moderator, the participants, and for the committee to make these events come through. That's all from us. Once again, thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Have a wonderful day, everyone.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.